Good morning, ma'am. Good morning, Shushmita, ma'am.
Good morning.
A very warm welcome from us. Please wait for a while. We are going to start the session in a while.
Sure, thanks.
Good morning, ma'am. Should I start?
Yes, sure.
Okay, ma'am. A very good morning, everyone. It gives me immense pleasure to extend to you a very warm welcome on behalf of Department of Ecological Studies, International Center for Ecological Engineering, and Department of Food and Nutrition, who has clubbed all of us for International Conference on integrated approaches towards sustainable management of environment for safe food, nutrition, and improved health. I am Shaptadipa Mojumdar and going to be today's host. I appreciate you taking time of your busy schedule to join us today. We hope it will be an informative and a fruitful session. At first, we are going to welcome Dr. Shushmita Shil. Shushmita Shil is the Assistant Professor of Department of Pharmacology, Nebraska Medical Center, USA. She has completed BSc, MSc, and PhD from Calcutta University and postdoc from University of Nebraska Medical Center, USA. She has more than 35 publications so far and guided 3 PhD students. She is a member of editorial board of medicines, current trained in pharmacology and clinical trials, Vessels Plus. Welcome, ma'am.
Thank you, Saptadipa, for the kind introduction. So, can I share my screen now?
Yes, yes.
Yes, ma'am. Yes, ma'am.
Okay, is the screen visible?
Yes, yes, it is visible.
Okay. Okay, at the onset, I would like to thank actually Doctors Manna and Bhakto for the kind invitation. So it's a pleasure to be here. And today I'm going to briefly talk on the research work that's going on in our lab. And I will be mostly focusing on one of our projects, which is surrounding astrocytes and astrocyte derived extracellular vesicles in HIV. So, what I will be talking today is how HIV can cause neurological disorders, the mechanistic role of astrocyte, which is all of you may know that this is one of the major CNS cell type in the brain, and specifically not only astrocyte but one of its secreted component, astrocyte derived extracellular vesicle, how it can cause neuronal injury, and finally how we are targeting it to develop a drug. So, a brief introduction before going to our main results section. So, we all know that HIV is one of the major causes of death in the world. And at present, around 37 million people are suffering on it, and around 27 million people are receiving the antiretrovirals. So, before the antiretrovirals came into play, AIDS was supposed to be a death sentence. But with the antiretroviral therapy, there have been a drastic difference in the life of HIV infected patients. In fact, there has been a lowering of the viral status from 2000 to 2020. You can see that people living with HIV have drastically increased in numbers, almost 30 million people living with HIV with lower viral load in the year 2020. So now, if the viral load is low, then what can be other problems? So these people who are infected with HIV, they do develop age-related disorders. And one of the major disorders are the neurological disorders that we see in this HIV patients. And that basically we term as HIV associated neurological disorder or HAND or neuro HIV. So, like our lab or a group of labs in our department, we are interested to figure out what is the reason behind this neurological disorders that we see in the HIV patients who are on antiretroviral therapy. So, what happens is that even when the viral load is down, the HIV proteins, like the HIV tag proteins, like keep on accumulating in the brain and in different other organs of the body. And this appears to be very neurotoxic because that causes loss of memory. In total, actually, it causes cognitive function deficit. So we tried to figure out how we can address this issue because this is kind of a big issue for the HIV infected population all over the world and especially in the United States. So, one of the major conditions of this HIV associated neurological disorders is the development of Alzheimer's like pathology. Now, what is Alzheimer's? Alzheimer's, as we know, is a neurodegenerative disease. And in Alzheimer patients, we do see a marked decline in cognitive functions. And here we wanted to figure out if the pathways that regulate this Alzheimer's disease is also regulating the HIV associated neurological disorders. Now I will be showing you some of the eminent scientists who have been working in this field for a very long time, and they have shown that in patients like HIV infected patients who are on antiretroviral therapy, they do have Alzheimer's proteins in their brain, in their CSF, as well as plasma. And what are those proteins? They are like the toxic amyloid beta proteins, the phosphotau proteins that can cause the neurological disorders. And especially, it has been seen that the HIV TAT protein, which I already mentioned, remains in the brain and other organs of the body, even in the presence of the antiretroviral therapy, do contribute to the process of neurological disorders. And there have been scientists who have shown that this can also lead to generation of neurotoxic amyloid beta, which can be produced by the neurons and the endothelial cells in the brain. So, we were basically not interested in neurons nor on the endothelial cells, but we were basically interested on the astrocytes. Now, these are glial cell population, and these are very well known cells for contributing to the inflammatory process in the brain. But we, for the first time, have shown that these glial cells, known as astrocytes, can produce the amyloid proteins in response to the HIV TAT protein, and that can increase the toxicity in the brain. So, from this background, we came up with the hypothesis like if HIV TAT protein can induce Alzheimer's like pathology, what can be the role of astrocytes and astrocyte derived extracellular vesicles in this process? So, before going into the results sections, there are a few questions that we asked before starting the study. So, the first question was that whether astrocytes can produce amyloids in the CNS. Next question, if yes, what is the mechanism that is happening? Third, it is very important to see in any of research based studies what is happening in our in vivo system. And for this case, we have used monkey models as well as HIV infected patient samples to prove our hypothesis. And finally, how the role of astrocyte derived extracellular vesicles can play an important role in disease progression and how it is important as biomarkers. So, I would like to briefly show you what I am actually going to talk about. So, in Alzheimer's disease, what happens is that there is a transmembrane protein which is called the amyloid precursor protein or the APP. And under pathological conditions or under disease condition, this is cleaved by the base 1 enzyme and the gamma secretase enzyme, and that can cause the secretion of the neurotoxic A beta protein. Now, more of it, which comes out of the cell, can accumulate with each other to form the amyloid plaques. Now, these amyloid plaques are neurotoxic by itself. That can directly lead to cognitive impairment. And in addition, that can cause neuroinflammation in the brain, which can again lead to cognitive impairment and progress the neurodegeneration in the brain. So, this is the total mechanism that happens in Alzheimer's disease. And we really wanted to see whether this phenomenon was also happening in our HIV brain samples because we do see similar symptoms in these patients, just like Alzheimer's disease patients, like loss of memory. So, what we have done is that we have taken human primary astrocytes in our in vitro culture and we have exposed it with different doses of TAT protein, HIV TAT protein in culture. And we have seen in a dose dependent manner all of the amyloid markers like the A beta, the APP, and the cleaving enzyme base 1, all of them were significantly upregulated. And as you can see here, base 1 is an enzyme. So it is, in addition to look into its expression, it is important to look into its activity as well. And here we could see that the base 1 enzyme activity was increased as well. And through qPCR analysis, we have seen not only the activity, but also the mRNA expression of this amyloid precursor protein and the base 1 enzyme was significantly upregulated. And the most interesting part was that after formation of this amyloids in the cells, we could see in the cell supernatants that this amyloids were secreted by ELISA. So, this part of the result showed us that when we expose the astrocytes to HIV TAT protein, it can increase the markers of AD, and that can additionally be released in the supernatant. So next the question comes is that what or the mechanism how these amyloids are getting produced in these cells. So so next we did a bioinformatic analysis to look into the promoter region of the base 1 enzyme and what can be the factors that can target the promoter of the base 1 enzyme. So we have figured out two important components, or I would say two targets that can control the expression of the base 1 enzyme. The first was a transcription factor known as the hypoxia inducible factor 1 alpha. And in the same astrocytes, we did see in upon exposure to TAT protein, there was a significant increase in the HIF1 alpha, and it was not only in a dose dependent manner but also in a time dependent manner. So and next we have seen, since this is a transcription factor, the way the transcription factor acts is it goes into the nucleus and it binds to the promoter of its target gene and either induces or suppresses its expression. So here it was important to see whether this HIF1 alpha can be seen in the nuclear component. So from the cells, we have divided the nucleus and the cytoplasm. And in the nuclear component, we have seen upon response to TAD that the HIF1 alpha accumulation was happening. And in the same set of cells, we have seen in the cytoplasmic compartment, there was a decrease in the HIF1 alpha. And when we have done immunohistochemistry study with an overexpressing plasmid, again, you can see that upon response to TAT protein, there was an increase in the HIF1 alpha in the nuclear component mainly. Our second target of interest was a long non coding RNA known as base 1 antisense. So basically, coding RNAs can be transformed into protein in our system. But for a long time, we thought that the portions which are cut out and we thought those are junk RNAs and doesn't have functions, those are called long non coding RNAs. And it is of interest that this long non-coding RNAs, although they can't help in protein synthesis directly, they do have multitude of different other functions. And here, such type of an RNA is a base 1 antisense. And here we have seen in our system in the cells that the base 1 antisense was also time dependently upregulated, and they were mostly confined in the nuclear component of the cell. And again, this is an in situ hybridization by which we have seen in the SIV infected monkey brain, there was an increase of this long non coding RNA in compared to the saline group, which is, you can see this by the red signal. And the white is basically the marker for astrocyte, which is the GFAP. So they were co localizing with that. So, which means the astrocytes are basically producing this long non-coding RNA in the brain of these monkeys which are infected with SIV. So next we wanted to figure out how all of these components like the HIF1 alpha, the base 1 antisense, and these amyloid components, they were related to each other. So the strategy we have taken is that first we have silenced the HIF1 alpha gene in our system with a specific targeted siRNA. And when we silence the HIF1 alpha gene, we could see that the mRNA expression of the long non-coding RNA was going down, the secreted A beta was going down, and in the cells, the A beta, the base 1, as well as HIF1 alpha was downregulated. So this made us believe that HIF1 alpha may be the master regulator that is regulating the long non-coding RNA, the base 1, as well as the amyloids. So but we have to always in science we have to see the reproducibility of our data. So we next basically silenced the base 1 alpha gene, base 1 antisense gene, and with the silencing of base 1 antisense we saw that the base 1 and the amyloids were inhibited, but the HIF1 alpha still remain upregulated in our system, both in the mRNA level as well as in the protein level. And this made us believe that HIF1 alpha is the master regulator that can regulate the long non-coding RNA based 1 antisense, and that can cause the generation of the amyloid proteins in our system. So now that we have found that this HIF1 alpha and the base 1 antisense are basically cross talking with each other, and this has not been reported earlier in literature. So we were interested to see what was their strength of association, basically, which means how these two components were talking with each other. So to look into that, what we have done is that we have done an RNA immunoprecipitation assay. So here what we have done, we have treated our cells with the TAT protein, and we have pulled down the HIF1 alpha bound all the RNAs with a HIF1 alpha antibody. And in that enriched component, we have seen that the long non-coding RNA base 1 antisense was significantly upregulated in response to TAT protein. So, which made us believe that the HIF1 alpha and the base 1 antisense were actually present in the same complex. So, the next thing was to understand how both of this component can regulate the generation of the A beta. So next we performed an chromatin immunoprecipitation assay, and without going into much detailing of the procedure, so whatever results we got from the chip assay, we found that basically both of this component, the HIF1 alpha and the base 1 antisense, was binding to the promoter of this enzyme base 1 and can regulate its expression. So here we found a novel function of both of this complex on the base 1 generation. So next we wanted to see whether both of these components were directly associated with each other, or it was through some other components that they have shown a complex. So what we have done was that next we have done another type of analysis where we can see a simulated structure where we saw that the HIF1 alpha, which is represented in blue, and the base 1 antisense, which is represented in pink, whether they have any potential binding sites or not. And when we formulated this structure, and we have seen that both do have some common points of interaction. So it was like, so this basically gave us an indication that both of these components were actually interacting with each other. So next we had to see this experimentally whether this is happening or not. So we performed another assay called the electrophoretic mobility shift assay, whereby we have synthesized this base 1 antisense RNA in our lab, as well as we have taken HIF1 overexpressing cell extract, and we have put them together in our in vitro system. And you can see here, with increasing doses of base 1 antisense RNA, there was increased binding with the HIF1 alpha. So, which basically means that the HIF1 alpha and this base 1 antisense are directly associated with each other in our system, that is, in the astrocytes, in response to TAT treatment. So now, after figuring out the detailed mechanism how HIV TAT can induce astrocytic amyloidosis, the next question we had was that whether we see the same phenomenon in the SIV infected macaque brains or in the HIV infected patient samples. So here we have taken macaques where we have infected with a SIV virus for 12 months. And after that, we sacrificed animals, and we have taken the different brain regions of these macaques, and we used it for our further experiments. And here in this figure, you can see we have done immunofluorescence staining of the A beta 42 along with the GFAP positive astrocytes, which is in green, and the A beta is in red, and the DAPI, those are markers of nucleus. And here you can see in different brain regions like the frontal cortex, parietal cortex, hippocampus, as well as basal ganglia, there was significant deposition of amyloids in comparison to the saline group, which basically made us believe that in these animals, of course, there was synthesis of amyloids happening, and not only synthesis of amyloids. As I showed you earlier, this amyloids, when gets released from the cell, can form amyloid plaques, and that's what we are seeing here. And just like this amyloid beta protein, we have seen other markers like the phospho-tau protein by Western blot analysis in the SIV infected macaque brain regions. And in the same brain regions, we saw there was an increase of the phospho-tau protein, which is again another hallmark of the Alzheimer's disease. And just like phospho-tau and A beta, we have also seen that the APP and other varieties of amyloids were also upregulated in the macaque brains. And for the interest of time, I'm not showing those detailed data here. So next we have done an RNA-seq analysis in the monkey brain regions, and there we have seen that in the SIV infected brain regions, there was significant upregulation of all of these amyloid proteins. Also, the base one is enzyme, the HIF1, so, the GFAP positive astrocyte, and the IBA1, which is a microglia specific marker. So we can see not only in the protein level, but also in the RNA level, the gene expression is being upregulated of our target genes and proteins of interest in the SIV infected macaque brains. So next, we will move on to see what is happening in the HIV infected brain section of the patients. So as I was telling earlier, that with the antiretrovirals in the HIV patient, the viral load definitely goes down. But the new complication in the situation is development of cognitive deficits in these patients with antiretrovirals. And there are different types of cognitive deficits observed in these patients, like the asymptomatic ones, where we do see pathological changes but not much of symptomology. Next, we see the mild neurocognitive disorder where the patients start having this memory loss and other type of cognitive deficits, and HIV associated dementia, which nowadays have really reduced a lot with the antiretrovirals to 2%. So mainly we will see the asymptomatic neurocognitive impairment and mild neurocognitive disorder in the HIV patients. And coming to the staging, you can see from stage 0 to stage 4, the patients almost become vegetative with no intellectual ability, no motor disability, and complete loss of memory function. So we have a brain bank here in US where we can request samples, and from there we have got these patients who are in the asymptomatic zone infected with HIV, and in the mild neurocognitive disorder zone infected with HIV, and patients HIV who are detected with Alzheimer's pathology. And here the control means the HIV negative group. And this all are male patients, and they are of the same age group, 40 to 60 years of age. So here we are showing in the frontal cortex brain region, you can see specifically both in the asymptomatic group and in the cognitive impaired group, there has been upregulation of the amyloid proteins as well as the enzyme as well as HIF1 alpha as well as another pathological marker, the phospho-tau protein. Now you can see how we are moving with this work. We started from cell culture in astrocytes, then we moved to macaque brain sections. Now we are seeing in human patients. So it is very important to verify all of this study in different model system to see the reproducibility of our data. And next, again, we have done the immunofluorescence study in the patient samples. So these are the in the front, the hippocampus brain region. And here you can see there was lot of deposition of this neurotoxic A beta protein, and they were co-localizing with the astrocytes. Again, making us believe that astrocytes are producing this amyloid proteins. And again, we did do an in situ hybridization where we have seen that the APP RNA, which is marked by the red signal, is co-localizing with the astrocytes in the brain, which is this green signal, green colored cells, and which basically showed us, like again, that the astrocytes are the producers of this amyloid proteins. And so our main theory was to check not only the amyloids but also its inducers. So there we have seen in the HIV brain patients, there have been increase in the long non-coding RNA base 1 antisense, and they were very well co-localizing with the GFAP positive astrocytes. So again showing that not only in the macaque sections or in the astrocytes in culture, but also in the human patient samples, the astrocytes are producing this long non-coding RNA. So basically, we came to the answer of our third question is whether there was any region-specific astrocytic amyloidosis in SIV and HIV, and the answer is yes. So the next we were interested to see what is happening in terms of what is the function of this neurotoxic amyloids that we are talking about for so long. So we will see what is the function next and how the astrocyte derived extracellular vesicles are playing a role in this process. But before going to that part, I want to summarize what I have said until now. So this is broadly the astrocytes, and this is the HIV TAT protein. When we are exposing the astrocytes with the HIV TAT protein, there is an upregulation of this transcription factor in the cells known as the HIF1 alpha, then which gets translocated in the nucleus of the cells. It binds to the long non-coding RNA link RNA base 1 antisense. This complex together can regulate the promoter of the base 1 enzyme and regulates its expression and cause RNA stabilization after transcription, and then that leads to slow production of base 1 protein for a long time, and this being an enzyme then cleaves the amyloid precursor protein to form the neurotoxic amyloids that comes out of the cells, and they can form amyloid plaques. Those are neurotoxic to the brain, and that in the next section we will see how it is leading to cognitive impairments. So, next, what I will be talking about, a brief background on that, I will be talking about exosomes or extracellular vesicles. So, these are double membrane structure vesicles that can be secreted out of the cells, and it can contain RNA, it can contain protein, and it can contain lipids and various other factors in them. And what is its importance? It can cause cellular crosstalk. Suppose it is coming out of the astrocytes, it can go to the neurons and it can transfer the astrocytic material from astrocytes to the neurons or any other cell types in the body. So, this exosomes we have targeted to see whether they are containing the amyloids which are produced in the astrocytes and whether they can be taken up by other cell types. And there have been literature where people have shown in Alzheimer's disease that A beta can be carried through these extracellular vesicles. And even in HIV patient blood samples, they have shown that these exosomes can carry these amyloid proteins, which are coming from the brain or neuron specific. So here, our main purpose was to show that the brain, which were producing the amyloids, whether they were carried through these extracellular vesicles and whether they're reaching in other parts of the body and can be targeted as a biomarker for whatever is going on in the brain of these patients. So what we have done is that we have taken next an HIV transgenic rat that is available in our department. And this rat, actually, they are not infected, but they act just like HIV patients who are taking antiretrovirals. That is, they have a lot of HIV proteins in them, but not the virus in actual. So we have taken those rats and taken out the brains of those rat, and we have isolated this extracellular vesicles. So this is kind of a long process, so I won't go again into the detailed methodology of that. But what we have found, our main finding is that you can see that this vesicles which are like isolated from the brain from the astrocytes, they were increased in number, not only in number. If you see their cargoes, the amyloid cargoes in them were extremely increased. That is interesting. But more interesting is that we saw similar phenomenon in the serum. So in the serum of these animals, we also saw that this astrocyte specific extracellular vesicles were increased in numbers, and they were containing amyloid cargoes. So why this is important? Because this can serve as a biomarker of a disease. So in a living patient, we actually cannot go and take out the brain and see what is going on. But to see or assess what is going on in the brain, we can definitely take the blood and take out the serum from it and can check this extracellular vesicles and whether they are having this amyloid proteins in them or not. And that can basically tell us what is going on in the brain in terms of neurological disorders or the cognitive impairments that we are observing. So this can serve as a biomarker, and this we have of course done in HIV transgenic rats, but in human patient samples, this work is ongoing. So, so the question that I raised before, that can astrocyte-derived extracellular vesicles can serve as a biomarker, and the answer to it is yes. So my next question is that, well, then what is the functional role of this astrocyte derived extracellular vesicles carrying the amyloid cargoes? So that will be my final question and answer for this talk. And so what we have done is that, so as I told, that this extracellular vesicles can be secreted through the cells, specifically the astrocyte. So we have taken the supernatant of the astrocyte growing culture, and we have isolated the extracellular vesicles. And this extracellular vesicles, then we did detailed characterization. Next, we have exposed the neurons with this extracellular vesicles, which we have taken from astrocyte culture, and we have seen what type of damage it can cause to the neurons. And as you can see here, this is basically in a brain. This is in the brain of the mice. We have tagged a neuron with an RFP plasmid, which kind of emits red color, and with help of a software, now we can see what type of branches are getting I mean what type of branches are getting shorter and what is getting longer, whether these neurons are losing any spines in them or not. So through which, through this detailed characterization of the neurons, we can answer the question what type of effect these amyloids or any other neurotoxic components are having on the neurons. So you can see this is a picture of the neurons, and these are basically the TX red labeled EVs from astrocytes, and this you can see can be taken up by the neurons after 24 hours of exposure. So this was our first thing to see. Next, we saw that this astrocyte derived extracellular vesicles, or which I will now term as ADEVs, was significantly upregulated in the TAT exposed group compared to the control group. And the most interesting part was that you can see here that these EVs contain the amyloid protein, the amyloid precursor protein, and the A beta protein, which we consider to be the neurotoxic one. And what we have done, we have exposed this ones on neurons in culture. And this, then we have seen, this is kind of the dendrites of the neurons. And you can see this in the control, there are the spines. And the spines are the point of contact between two neurons for proper impulse propagation. But you can see when the TAT ADEVs are exposed to the neurons, they are losing the spines. And this we have shown by the quantitative analysis as well. And we have also seen VGLUT1, which is a marker of excitatory neurotransmitter, which is basically required for proper impulse transmission in the brain, also getting significantly downregulated in exposure to the TAT ADEVs. So from this, we get an idea that this EVs, which are carrying amyloids in them, when we expose to normal neurons, that can cause a real damage to them. So that is the reason we can propose why we are seeing the neurological disorders in these HIV infected patients who are on antiretrovirals. And so next we wanted to see whether these amyloids that were coming out from the astrocytes, what type of neuronal injury it was doing, and whether when we are blocking the HIF1 alpha, so whether there is protection of this phenomenon or not. So, uh, sorry, any questions? Okay. So what you can see here is that these are my normal neurons, and when we expose with the TAT ADEVs, you can see all the dendrites and branches are getting highly reduced, which again means like the normal functioning of the neurons are getting affected. And of course, when we are silencing the HIF1 alpha gene, this is getting protected. And when we are looking into the spine density, again, specifically, we see that with exposure to TAD, the dendritic spines and the excitatory proteins are going down. But when we are silencing the HIF1 alpha gene, this is again getting protected. So, which basically tells us that HIF1 alpha is basically a target molecule that can be used for drug development. Next, what we did, we took this extracellular matrix and we injected in wild type mice, which is basically the naive mice in the hippocampus. And we have seen what is happening in terms of their cognitive impairments. And we have done a maze study with this mice, and we have seen memory, and we have also seen locomotor activity. The motor activity of these animals was significantly upregulated. And if you can see the brain structures in these animals, you can see I have done the immunohistochemistry staining. There was an increase of astrocyte activation, increase of microglial activation, decrease in the neuronal marker, presynaptic protein density protein 95, and also increase in amyloid pathology as well as in the cytokine level, which leads to neuroinflammation. So when these EVs, which are treated with TAT from the astrocytes, are injected in the brain, there is significant upregulation of the glial activation, the amyloid pathology, as well as neuroinflammation. So you can see when we are injecting these EVs in the, although we are injecting in the hippocampus, it is getting transported in different parts of the brain, not only in the brain, we can also see in the serum, and that can serve as a biomarker of the disease. So, I would summarize what I have talked about until now. So, Alzheimer's like pathology is one of the contributing factors to HIV associated neurological disorders that are observed in HIV patients who are taking antiretrovirals. The astrocyte derived extracellular vesicles carrying amyloid proteins can serve as peripheral biomarkers. This ADEV plays a significant role in disease progression and in causing cognitive impairments in the HIV patients. And the HIF1 alpha can be considered as a master regulator in the process of amyloidosis mediated these neurological disorders. And the significance of the study is based like this HIF1 alpha can be developed, can be targeted to develop therapeutics, not only for HIV, but also like our lab also works on drug abuse and also other neurodegenerative diseases where we see that the HIF1 alpha mediated amyloid pathway is very dominant. And the things that we are working on now, that we in our department, we have humanized mice. This mice basically have 70 to 80% of human cells, and we are actually developing a target to silence the HIF1 alpha in this mice and look at the HIV viral load and the HIV associated neuronal disorders. And I would not go much more details into that because this work is under consideration as a patent now. So, yes, that's pretty much it, what I wanted to share with you today. And thank you everyone for your patient hearing. And before wrapping up my talk, I would like to acknowledge a host lot of people with whom I have been working of course, all the lab members and the students and the funding agencies, the NIH, all my collaborators, specifically my mentor and our chair of the department and the NNTC from where we get our tissue, the human tissue. So, with that, thank you all. And if you have any questions, I am happy to answer.
Hello. Hello.
Yes. Hello.
So, yes, very good morning, ma'am. I am Dr. Bhakta speaking from the Department of Food and Nutrition, University of Kulani. Really, I am very much pleased to have your very nice presentation and illuminating presentation. It will give a lots information to our participant, not only the lot, but very new and very informative and very novel informations to our participants. So I think everybody have got sharp information from this lecture, and I request to participants, the session is now open for discussion. You can ask and interact with our very much valuable speaker, uh, Dr. Sushmita Sil. Please, participants.
Thank you, Dr. Bhakto. Yes, if any of you have any question, yes, Fardin.
Maybe not any questions. I think.
No, someone raised the hand.
Yes, please. Please ask some questions.
Fardin, you raised your hand. Do you have any question?
If you have any question, please ask. Maybe not. I think so. So, ma'am, really we are pleased and to have your time from your busy schedule during this period. And I hope you always extend your kind hand to collaborate with our department to flourish our
Sure,
new
sure.
department, yeah, establishing a very much premature juvenile stage. It is really very just 2 years and just third semester is going on. I hope your kind support will expect in future. Please, madam, thank you again for your nice presentation. Okay, madam.
Thank you. Thank you. Okay.
Thank you so much, Sushmita ma'am, for such an informative presentation. Have a good morning, everyone. I am Rupsha, student of Department of Food and Nutrition, University of Kalani. It's an immense pleasure for me to be a part of this session. With due respect, I would like to welcome Dr. Shayantan Rai. Dr. Shayantan Rai presently serves as a senior consultant and endocrinologist at Apollo Hospital, Kolkata. He is currently affiliated with Welcome Medicare, Bhuvanipur, Kolkata, West Bengal, and Jagannath Gupta Institute of Medical Science and Hospital, Kolkata, West Bengal. He holds a MBBS degree, MD in medicine, and DM in endocrinology. Dr. Shantan Rai has over 50 publications in national and international peer reviewed journals. He is a member of Endocrine Society of India and American Association of Clinical Endocrinologists. Additionally, he is also the author of RSSDI Yearbook of Diabetes 2017. Today, Dr. Shantan Rai sir here is with us and will be discussing on the topic vitamin D in health and disease, understanding of the traditional functions and new rules. Welcome, sir.
Thank you very much for this kind introduction. May I share the slides?
Yes, sir.
Is my screen visible?
Yes, yes, it is visible.
Okay. So may I start?
Yes, sir, you may start.
Oh, thank you. Thank you very much. So, good morning, everyone. At the outset, I must thank the organizing committee of University of Kalyani for inviting me and giving me the opportunity to deliver my talk in front of this August gathering today, although we are doing it on virtual platform. So my topic for the next half an hour or so will be vitamin D in health and disease, an insight into the traditional functions and some new roles. So as we know that vitamin D is unique among the vitamins in that human can synthesize it via the action of ultraviolet B radiation upon the skin. Additionally, its ability to act on specific target tissues via vitamin D receptor make its classification as a steroid hormone more appropriately. So if we look at this world map, as we are also going through a pandemic of COVID-19, this particular illustration is showing a vitamin D pandemic.
Extremely sorry
Vitamin,
to interrupt you, sir, but I think
yeah,
there is a technical issue in your network. The sound is
this,
not coming properly, sir.
just a
No,
minute.
but here the sound is coming. Akalika, we can hear it. I think the participants, can you hear? No.
Okay, ma'am.
Participants, one of you please respond
Yes,
whether.
ma'am. Everything is visible and audible.
Yes, yes.
Oh,
So,
Yes,
oh,
yes.
Akalika, we
Nothing
can
is
that's
broken,
fine.
see
ma'am.
something maybe issue from your side. Okay.
Yeah, that's fine. So, so if you look at this vitamin D deficiency pandemic, that means reported incidence. If you look at the reported incidence of vitamin D deficiency, which is defined as 25 hydroxy vitamin D level below 20 nanogram per ml across the globe in general population as well as pregnant women. So you will appreciate that a significant proportion of general population as well as pregnant women are having vitamin D deficiency. They are not at suboptimal level of vitamin D. So while vitamin D deficiency, the recognized problem in some northern latitude countries, recent observations have shown that even in sunny countries like our own country, like Australia, vitamin D deficiency may be more prevalent than earlier we thought. Now, if we look at this scale, that is the opinions on threshold for vitamin D status as defined by what I have just mentioned, 25 hydroxy vitamin D concentration, it has been reported that there is an inverse relationship with serum PTH levels and 25 hydroxy vitamin D. That means with serum parathyroid hormone and 25 hydroxy vitamin D. And that parathyroid hormone level usually begin to plateau at approximately 30 nanogram per ml. That's why most of the contemporary guidelines advocated that concentrations between 20 and 50 nanogram per ml appear to be safe and sufficient in the general population from skeletal health perspective. On the right panel, you can see that vitamin D acting through vitamin D receptor may have various function in different systems of our body. We will come into details of that. Now, one important question. Now, we know that uh, we take vitamin D supplements. Often, doctors prescribe vitamin D supplements either with calcium preparation for daily consumption, sometimes a weekly large dose of vitamin D, and also sometimes a monthly dose of 60,000 unit international unit of vitamin D. Now the question is that where from the vitamin D3 in supplements is derived? Any answer from the participants? Any idea from where vitamin D3 is derived to prepare these supplements what we often take? And in during this pandemic, many of us have taken vitamin D3 supplements. So it usually derived from either fish oil or from the cholesterol obtained from lanolin in sheep's wool. And vitamin D2, that is argocalciferol, D3 is cholecalciferol, that is usually produced commercially from ultraviolet irradiated yeast. And argocalciferol, that is vitamin D2, is present in mushrooms. That could be an important dietary source of vitamin D2. Those mushrooms exposed to sunlight or ultraviolet B radiation, and also in sun-dried mushrooms. So coming to this important area of production of vitamin D, endogenous production of vitamin D in the skin during exposure to ultraviolet radiation in sunlight, which actually starts the synthesis of cholesterol, means cholecalciferol from 7-dehydrocholesterol. Then this pre-vitamin D3, after binding to vitamin D binding protein, you can see here, it is presented to liver. And the dietary source of D3 and D2, which is only 20% of the vitamin D in the circulation, which comes to the circulation, 80% is synthesized in the skin under the effect of ultraviolet B radiation, transported to liver, where it is
Excuse me, sir.
hydroxylated.
Sir, your
Yeah,
slide is not changing. It's just showing the first slide only.
it's changing. Uh, one thing we can do, I should stop my video.
Yes, so it's showing slide number 4.
Yeah, just a minute. Is this now visible?
So slide number 4 is visible now.
I think if it is shown in the slideshow mode, all the slides will be visible.
I have already put it in slideshow.
So then, before sharing, I think it has to be the total screen share. Then the slideshow will be
Just a
shown
minute.
here.
Just a minute. Stop sharing first. Then. Is the slides visible?
I think only one slide is visible. Again, the slideshow mode is not coming.
Slideshow is not coming.
Before sharing the screen, sir, if you go to the total screen share, computer, total computer, then you go to that particular file and.
Okay, I understand. I have to do that. Screen.
So you just change the slide and I'll just tell you whether it's changing or not.
I just see it. I have already shared. Actually, if I share the entire screen, my slides are not coming. That's why I'm sharing the window.
Oh. So now you change the slide, I'll just tell you whether it's changing or not.
I'm changing the slide.
But here it's showing only the first slide, sir.
This is the problem. Uh, mm.
Sir, after sharing the whole screen, you just click on the presentation, the PPT, and after that you
When
just
you
press
sharing,
publish.
if I stop present here, but when I am sharing the entire screen, I could not see my presentation there.
In
Only,
the down
only I.
the bar that is there down, you first open the slide, sir, and minimize it. After that, you
Yeah.
share the entire screen, and from the bottom, open the slide
Okay,
after
okay,
that.
okay, okay. So, like I'm sharing my entire screen now, but it's not showing any slide here.
But the slide, you first open the slide and then share your entire screen. Open the slide, minimize it, and then share the entire screen. And then from the down bar, you just click on the presentation, it will open.
Okay.
Dr. Santon, if you want, you can
Yeah.
share to me
That will,
your slide.
I think
Send to
that will be
me.
better. You send the file to
Then I
Dr.
have to
Manna.
send it to through email.
Yes, yes, yes.
Yes, sir. You email it to
I am
Dr.
sharing on behalf of you.
Okay, no
He
problem.
will
I'm
be
sharing.
presenting it, and you can just see
I
the.
Send
am
it to,
sharing
yes,
the
yes. You
slide for
send it
you.
to me.
No problem. No problem. Yeah, I have shared.
In which email you have sent?
So the email I received from your end.
HOD food.
Yeah.
Yes, yes, yes. I got your slide.
Oh, fine.
Okay, just a minute. Sir. Is it visible to you, sir?
Yeah, it's visible. You can now put it in the full screen mode.
Yes.
So the next slide, I think. Now it's visible. Okay.
Okay.
So if we look at the reported incidence of vitamin D deficiency, defined as 25 hydroxy D level below 20 nanogram per ml across the globe in general population and also in the pregnant women, you appreciate that a significant proportion of population are having a suboptimal level of vitamin D. And while vitamin D deficiency is a recognized problem in some northern latitude countries, recent studies have shown that even in sunny countries like ours or in Australia, vitamin deficiency may be more prevalent than earlier thought. Next slide. To the left, the scale of opinions on thresholds for vitamin D status, defined by serum 25 hydroxy D concentrations, and it had been reported that there is an inverse relationship with serum parathyroid hormone and 25 hydroxy D levels, and
Just a minute,
that
sir. Just a minute,
PTH,
sir.
no problem.
Just a minute, sir.
That PTH levels actually begin to plateau at approximately 30 nanogram per ml. That's why contemporary guidelines advocated that concentrations between 20 to 50 nanogram per ml appears to be safe and sufficient for general population in terms of skeletal health. On the right, the major either potential or a real target systems for vitamin D actions. Although vitamin D is well known for its role in bone health, however, discovery of vitamin D receptors on a wide variety of tissue types has opened up the roles for vitamin D far beyond the traditional role in bone health. Next slide. So, as we know that we take vitamin D supplements, but question is vitamin D3 derived from what? I think one source is fish oil and another one is cholesterol obtained from lanolin in sheep's wool. And vitamin D2, that is argocalciferol, is produced commercially from UV irradiated yeast, also present in sun-dried mushrooms, which could be a good dietary source for vitamin D2. Next slide. You can skip this slide, just go to the next slide. This slide focuses on endogenous production of vitamin D in the skin during exposure to ultraviolet radiation in sunlight. It starts with synthesis of cholecalciferol, vitamin D3 from 7-dehydrocholesterol. After binding to vitamin D protein, the dietary source of D2 or D3, which is 20%, 80% is from skin synthesis, transported to liver. They're hydroxylated to 25 hydroxy D, that is calcidiol, which can be stored in the liver as well. And 25 hydroxy D is the major fraction of vitamin D in the body. Most of it circulates in the bloodstream with some storage in the adipose tissue. That means in fat. And synthesis of active hormone, that is 125 dihydroxyvitamin D in the kidney, that occurs through activation of, you can see here, the 1-alpha hydroxylation, which is tightly regulated by parathyroid hormone and fibroblast growth factor 23. A few other cells like intestinal cells, pancreas, prostate, and immune cells like macrophage can also hydroxylate 25 hydroxy D to the active form. That is actually an autocrine type of action. And this active form of vitamin D on your right panel enters the cell and binds to a nuclear receptor, that is vitamin D receptor, that is a nuclear hormone receptor, and thereby regulate the gene transcription, that is the genomic pathway of action. There is another pathway that 125 dihydroxy D may also activate, that is a minor part in this non-genomic pathway. Next slide. Next slide, please. So this is showing an overview of metabolic pathways with different ranges of vitamin D status, but depending on the blood level of vitamin D, the production of active form and also the degradation to inactive form, this catabolism and anabolism, depends on the level of vitamin D available in the circulation, and in that way the activating enzyme 1-alpha hydroxylase or the inactivating enzyme 24 hydroxylase becomes more functional in that fashion. Next slide. So if we look at the potential skeletal and extraskeletal target tissues and effect of this vitamin D endocrine system, robust evidence we have on calcium and bone homeostasis, on the immune system, and on hormone secretion. Whether in other systems where vitamin D receptors are present, like heart or blood vessels, muscles, skin, brain, and of course lung, the role of vitamin D is still not clear. In the subsequent part of the presentation, I shall take you through the skeletal and non-skeletal effects of vitamin D. Next slide. So we know that vitamin D is essential for skeletal health. That's traditional teaching. Next slide. If we look at vitamin D receptor signaling and intestinal calcium transport, in order to maintain a normal calcium levels, calcium is absorbed from the intestine and reabsorbed in the kidney, depending on the need of one's system. Now, adequate intestinal calcium absorption is required to acquire and preserve the bone mass as well as the bone quality. And 125 dihydroxy D is considered the primary factor to regulate this intestinal calcium transport. And a transcellular pathway that is actually controlled by this active form of vitamin D that transports calcium in an energy demanding process. And when intestinal calcium absorption is not sufficient, renal reabsorption will be more. And when that is also insufficient, your bone will serve as an additional pool of calcium during the negative calcium balance. Next slide. If we look at the role of vitamin D receptor in the kidney, next slide, please. The kidney can quickly adapt calcium reabsorption when intestinal calcium absorption is decreased. The regulation of calcium absorption occurs primarily in the distal nephron, where active vitamin D stimulates the active transcellular pathway. And we have seen that in VGR null mice, renal calcium reabsorption is impaired, as shown by the inappropriately high urinary calcium levels in presence of hypocalcemia. Next slide. Now, on the left panel, if you look at the model of normal calcium balance vis-a-vis a scheme of negative calcium balance. Next slide, please. Now, on the left side, this is a model of normal calcium balance. What happens when normal serum vitamin D levels promote intestinal calcium absorption? You can see here when dietary calcium supply is low, normal to normal, and stimulate also renal calcium reabsorption in the distal tubules. And this pathways deliver sufficient calcium for adequate bone matrix mineralization. Now, in VDR action in mature osteoblasts is anti-catabolic. That means it prevents bone resorption by increasing the LRP5 expression and decreasing the rank ligand expression. So, two mechanisms that contribute to the transfer of calcium from bone when there is negative calcium balance. That is in response to the increased 125 dihydroxy D and PTH levels comes here to act. One is increase in bone resorption caused by a high rank ligand. You can see here on the normal calcium balance, there was down regulation of rank. But when there is negative calcium balance, there is up regulation of this rank ligand. And also a high rank OPG, osteoproterogen ratio of bone matrix mineralization is then suppressed. Bone actually becomes the source of calcium to maintain the blood level of calcium. Next slide. Now, if you look at the paracrine and endocrine role of vitamin D receptor in bone, there is a temporary role of vitamin D receptor in chondrocytes. That's a paracrine function. Vitamin D receptor signaling in chondrocytes has no cell autonomous role in the growth plate of the bone. During a positive calcium balance, when sufficient calcium is delivered to the bone, 125 dihydroxy signaling can regulate locally the bone metabolism. But this seems to differ with the differentiation stage of the osteogenic cells. That means VDR action in osteogenic cells, that is bone cells, dependent on the calcium balance, what I have shown, normal and negative calcium balance, and also the cell differentiation stage. When there is mature bone, then it will prevent catabolic action or bone resorption. And there is a subtle role of VDR in the parathyroid as well. So when there is negative calcium balance, that is during deficient intestinal calcium transport, actually the body will prioritize the preservation of constant serum calcium level over bone mass and quality. Although there is a role of VDR in parathyroid, hypocalcemia, that is low level of calcium in the blood, is actually the major driver of hyperparathyroidism. And VDR has only limited function in parathyroid physiology. Next slide. Now we come to the implication to human disease. Next slide. So one important disease is become rickets. If we look the role of vitamin D in calcium homeostasis and bone metabolism, the bone pathology of children lack a vitamin D action either because of low endogenous vitamin D synthesis or a low dietary vitamin D intake. but this can easily be prevented or cured with vitamin D supplementation. Now, in the adult counterpart, osteomalacia, a bone pathology persisting nowadays that occurs after the closure of growth plate, that is characterized clinically by bone fragility and milkman loser fracture areas. If we take a radiograph to see, now we know that severe vitamin D deficiency is the cause of nutritional rickets and is existed as a true endemic disease. And it is still endemic in the Middle East and Gulf states, northern India, and northern China. However, clinical vitamin D related osteomalacia is usually found in subjects with explicit deprived access to UVB radiation and in patients with impaired intestinal absorption of vitamin D as a part of intestinal disorders of fat absorption, because it's a fat soluble vitamin that can happen with some inflammatory or autoimmune bowel disease or after bariatric or metabolic surgery, which is often performed in very morbidly obese individuals. What happens? Vitamin D deficiency actually impairs the intestinal absorption and renal reabsorption. And due to a very low active form, the transfer of calcium from bone to serum is also limited here. So the increased PTH levels now will enhance the bone remodeling, and the low serum calcium levels will interfere with matrix mineralization, resulting in an increased osteoid formation, and that will actually lead to increase in bone fragility. So these bones are more prone to fracture. Next slide. Osteoporosis, the role of vitamin D supplementation. Now, when there is less severe vitamin D deficiency, is also associated with increased bone turnover, and that can accelerate bone loss. But that does not prove the causality, as a low vitamin D status may also be related to osteoporosis due to some unrelated confounding factors. And the serum concentration of 25 hydroxy D that is required to obtain a positive effect on fracture risk is still disputed. We do not know whether levels higher than 30 nanogram would be better than 20 nanogram at population level. This is still unsettled. And most recent guidelines recommend a daily vitamin D supplementation of about 800 international units, always combined with a good calcium intake, that is nearly 1000 milligram per day, either from diet as well as from supplement in older adults who had a high risk of or having a documented vitamin D deficiency, because it has been seen that a combined calcium and vitamin D supplementation in older adults, especially those with poor vitamin D status, might decrease the risk of hip fractures and other major osteoporotic fractures by about 20%. Next slide. So as a whole, vitamin D deficiency as a risk factor for skeletal health. Vitamin D is generally considered good for bone because it can prevent or cure nutritional rickets and osteomalacia. Vitamin D can be bad for bone, what I have already shown, that in case of severe calcium deficiency, high serum concentration of 125 hydroxy D can stimulate bone resorption, that is the rank ligand mediated action. And from clinical point of view, vitamin D requirements in adolescents, pregnant women, and other young adults is less well defined. Next slide. So probably if we summarize that vitamin D has no direct effect on bone. Next slide. The effect is actually largely mediated by increasing calcium absorption from the small intestine, primarily in the jejunum, and for a subset of population, still we don't have a well-defined criteria for vitamin D requirement. Next slide. So if we look at the risk factors of low vitamin D status, you will appreciate that sunlight exposure, since it the major source of vitamin D for most children and adults, we should not uh, forget this, that very few foods naturally contain vitamin D. So for vitamin D synthesis, the only source is adequate sun exposure. So inadequate sun exposure and the low ambient UV radiation level is most important factor for low 25 hydroxy vitamin D levels. And pregnant women are more frequently have a poor vitamin D status than non-pregnant women of the same age. So that also have an implication to replenish vitamin D store during pregnancy. Next slide. So in this context, human breast milk contains how much vitamin D and calcium? It contains approximately only 20 IU per liter. Next. And 25 to 30 milligram per DL. This is from Chinese data. So that's why exclusively breastfed infants are more prone to vitamin D deficiency. And approximately 20% of the maternal vitamin D is transferred to the infant through milk. So that's why the Endocrine Society recommended a daily intake of above 4,000 international units of vitamin D for the lactating mother to satisfy the requirement of vitamin D for breastfed infants. And for your information, egg yolk also contains vitamin D, that is around 20 international units. Next slide. Now, what is the safe threshold to prevent rickets and osteomalacia? That is a 25 hydroxy D level of more than 10 to 12 nanogram per ml. So below this level, there is high risk of osteomalacia. So this concentration corresponds to the threshold below which intestinal absorption of calcium is impaired. And that's the important of this particular threshold value. Next slide is the last part of the presentation. That is the non-classical effect of vitamin D. Next slide. This is again extraskeletal effects of vitamin D continue to be debated. Now we know that many extraskeletal sites or tissues like heart, blood vessels, endocrine glands like pancreas, adrenal, thyroid express the vitamin D receptor as well as both 1 alpha and 25 hydroxylase enzyme. And this system does not appear to be regulated by conventional calcium and phosphorus metabolizing hormones like PTH or FGF23. And vitamin D acts generally to promote cellular maturation and differentiation while inhibiting cellular proliferation. So that could have an implication for cancer prevention. And several genes playing important roles in the cardiovascular system are targets of vitamin D signaling. Next slide. So we will discuss on these three important health outcomes: effect of vitamin D on type 2 diabetes mellitus. So what is evidence we have? The evidence From many observational studies suggest a link between low vitamin D status and risk for development of type 2 diabetes. However, large-scale Mendelian randomization studies and randomized controlled trials are convergent and do not support the use of vitamin D supplementation for the prevention of type 2 diabetes. So we need additional studies and more in-depth analysis of existing studies to validate the findings whether vitamin D supplementation could prevent the onset or progression of type 2 diabetes from a pre-diabetes status. Next slide. Strong preclinical data exist that link vitamin D with cell cycle control and cancer. Many observational studies have associated poor vitamin D status with risk of cancer or poor prognosis. Vitamin D supplementation in vitamin D repeat adults do not actually change cancer risk. Evidence from RCTs and in line with prior studies and Mendelian randomization results, follow up is an important issue. If we take a longer follow up, that is more than 4 years, we can see there is probably a role of vitamin D in preventing the incidence of certain cancers. So a link between vitamin D status and cancer incidence or mortality, we cannot exclude. But the thing is that it will be very difficult to verify. Next slide. Regarding cardiovascular events, we have convergent evidence from RCTs and Mendelian randomization studies that vitamin D supplementation does not reduce the risk of cardiovascular disease. We have few mega trials in last 3-4 years that also clearly confirmed the lack of benefit of vitamin D supplementation, in particularly in adults who are having sufficient vitamin D. Next slide. So in view of this current pandemic, little discussion on vitamin D and immune system. Next slide. The majority of immune cells, including T and B lymphocyte and antigen presenting cells like macrophages, express vitamin D receptor. That suggests a physiological role of vitamin D in regulating the immune response. So coming to the very important autoimmune diseases, in vitro, means outside body system, 125 dihydroxy vitamin D is a powerful immunomodulator. It is involved in regulating the innate immune system. The adaptive immune system is usually downregulated by active vitamin D. And therefore, vitamin D deficiency might predispose to autoimmune diseases. Observational studies have suggested this effect might also apply to humans as well, but too few intervention studies have been conducted to evaluate this, and no major randomized trials have addressed the possible primary or secondary prevention of the major human autoimmune disease. So currently, strong evidence exists that support a causal association between genetically low serum 25 hydroxy D levels with an increased risk of multiple sclerosis, which is a neurological disorder. Additionally, some epidemiological studies have also demonstrated that vitamin D supplements in infancy could reduce the risk of developing type 1 diabetes, which is again an autoimmune disease which involves the pancreatic secretion of insulin. Next slide. So infection, insufficiency of vitamin D that we know more frequently occurring during winter has been suggested to contribute the seasonal nature of flu-like infections in winter seasons. However, published data for this remains debated. Some studies suggest that specific polymorphisms of the gene encoding the VTR are responsible for increased sensitivity to respiratory infections. When we treat with vitamin D, the epithelial cells increase the levels of toll-like receptor, co-receptor CD14 and cathelicidin. Cathelicidin is a peptide that carries a broad spectrum antimicrobial activity. So these antimicrobial peptides may have some antiviral effects as well. So for production of these peptides, vitamin D receptor activation in the epithelium is important. And we know that vitamin D receptor is present in the lung epithelium. Next slide. Probably the last part, vitamin D and respiratory effects. Next slide. So proposed effects on the respiratory system, I have just mentioned the enhanced antimicrobial peptides, regulation of the inflammatory response, regulation of airway remodeling, and improving the muscle strength. Next slide. So, what role of vitamin D in prevention of respiratory infections? It is believed that vitamin D increases the production of natural antibodies. Vitamin D is also known to strengthen the immunity by inducing monocyte differentiation and inhibiting lymphocyte proliferation. It also postulated that vitamin D enhances the phagocytic activity of macrophages. And in macrophages, there are vitamin D receptors and the activating system 1 alpha hydroxylase we have in the macrophages. So vitamin D supplementation could decrease the events related to respiratory tract infection. There is need of more well-controlled trials to reach a certain conclusion. Next slide. So vitamin D and COVID-19. Now, next slide, please. Enormous health implications of the COVID-19 pandemic that caused by the worldwide spread of this SARS-CoV-2 virus, given that the link between vitamin D status and COVID-19 is very, very important topic of research. We know that the innate immune system is stimulated by 125 dihydroxy D, and this is in line with a decreased risk of respiratory infections with vitamin D supplementations in individuals with vitamin D deficiency. A possible link with poor vitamin D status and the risk of severity of COVID-19 has received great attention. It has been found that lower serum concentration of vitamin D were associated with greater mortality, greater need for intensive care treatment, and increased severity of illness in general compared with a better vitamin D status. However, these findings was based on observational studies. That's why the link between vitamin D status and COVID-19 is still unsettled. But many ongoing trials are there that may clarify this question. Next slide. So coming to the last slide for take home points. Now about a 1/3 of world population lives with suboptimal, that is below 20 nanogram per ml of serum vitamin D concentration. So that is a huge prevalence of vitamin D insufficiency in general population. So a few hints have emerged that vitamin D supplementation might have some extraskeletal benefits, but that is in people with vitamin D deficiency, who have some documented vitamin D deficiency. No reason exists at present to recommend vitamin D supplementation of already vitamin D replete individuals. Beneficial effects of combined supplementation of vitamin D and calcium in older adults with poor vitamin D status. We have evidence from studies that could reduce the risk of falls and fractures. Ongoing studies might help to provide a better understanding of who could be benefited from vitamin D supplementation in terms of extraskeletal benefit, that is reduced progression to type 2 diabetes from pre-diabetes, or decreased number of infections, improvement in lung function, and decreased incidence of cancer or overall mortality. So thank you very much, and extremely sorry for the technical glitch that happened at the initial part of the presentation. Now I would like to take questions if there is any. Sure. Now, there are some proposed mechanisms of vitamin D, since vitamin D has an important role as an immunomodulator. So that's why in mechanisms of most of the carcinogenesis, there is important role of immune system as well as cytokines and other mediators. So it is postulated that the mechanisms of carcinogenesis, probably vitamin D could act as an immunomodulator, as also it could prevent cellular proliferation, which is an important factor of spread of tumor. So if it can prevent cellular proliferation and it can have some effect on the mechanisms of carcinogenesis, like inhibition of cytokines and immunomodulation, that's why it is postulated that probably a sufficient vitamin D status could prevent incidence of certain cancers. No, in cardiovascular system, several genes are regulated by vitamin D receptor signaling. Although we don't have any specific data regarding that, however, it is again postulated that in patients with hypertension or in patients with atherosclerotic cardiovascular disease, probably a vitamin D deficiency could accelerate this hypertension or atherosclerosis related cardiovascular damage. That's why in some studies, it is also has been found that if we replete the vitamin D status who are actually having vitamin D deficiency, that could have some positive out in terms of cardiovascular outcomes, some positive role, although the specific mechanisms by which vitamin D could have a beneficial effect on cardiovascular outcomes is still not clearly known. That I don't know the answer specifically. But why this question is for? Means which context? Genomic metabolism. So as far as my knowledge goes, vitamin D metabolism or vitamin D catabolism, VDR is not related to that aryl hydrocarbon, but I'm not aware of any interaction of this two signaling. COVID-19 downregulates VDR, means vitamin D receptor signaling is downregulated by COVID-19. I have not, I don't have any data, or I'm not aware of any data that COVID-19 infection downregulate vitamin D signaling, or whether that means if COVID-19 infection downregulates vitamin D signaling, then it's very difficult to get the effect of vitamin D in those who are infected with COVID-19 virus. Because I don't have any, I'm not aware of any data whether COVID-19 downregulates vitamin D VDR signaling. And on the other hand, in any respiratory infection, either viral or bacterial infection, vitamin D has some role by through immune system and through production of antimicrobial peptides in reducing the severity of respiratory infections. And some studies have shown that uh, when patients are getting infected at that time, if they had sufficient vitamin D levels, that could be beneficial in terms of outcome of the infection. And in hospitalized patient, again, uh, admission vitamin D level have some important implications in outcome of these patients. Although we don't have any clear data from randomized control trials, only observational data. Sure, sure. What kidney problem? Renal stone. So, but yes, in during lactation, it is recommended that you have to take 1000 milligram of calcium and 800, at least 800 IU of vitamin D. But if you cannot take calcium supplementation, you can take calcium through dietary sources. And as far as I understand the renal issue, it's a fear of having kidney stones with taking calcium. But you should know that if we take vitamin D, ultimately it will increase the calcium level through intestinal absorption. So that's why actually there is no risk of kidney stone if we take a certain amount of calcium that is recommended uh, daily along with vitamin D. But if we take calcium through only dietary sources and receive supplementation of vitamin D, since vitamin D is not present commonly in the dietary sources, I think it's sufficient to maintain the calcium balance, because in lactation there is a very high risk of bone loss.
So my one is the last question. If possible, please take it.
Yeah.
I've already composed it in the chat box.
Uh,
Last one.
Arthritis.
Osteo.
Arthritis is an umbrella term. So arthritis could be a degenerative arthritis like osteoarthritis, or it could be an inflammatory arthritis like rheumatoid arthritis. And as far as uric acid is concerned, this is crystal arthropathy. Crystal arthropathy usually is not related to either inflammatory process or degenerative process. It is that monosodium urate crystals are deposited in the joints, mostly in the lower limb, like your first metatarsophalangeal joint of the great toe, where it actually, this crystals actually initiates the local inflammatory process that leads to swelling, pain that we call acute gout or podagra. And when there is a sudden decrease in level of urate in the body or uric acid in the body, that can also leads to precipitation of this urate crystals in certain joints and leads to this arthropathy that is known as crystal arthropathy or crystal related arthritis.
And what about in case of degenerative?
In degenerative, usually happens in, that's only because of weight bearing joints due to the aging process.
Thank
Yeah,
you.
thank you very much.
Yes, yes, yes. I think actually, I think she has left because just now she has informed me since her talk was at 11:20 and she waited for nearly more than half an hour and she has got some another talk at around 12:30, so she wouldn't be able to complete it. That's why I think she has left. So it will be better if we start our session. Okay. Shoptadipa, can you hear me? Hello, Shoptadipa? Yes, yes, Rita ma'am and Anshuman sir has joined, so we can begin our session. Yes, 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 Shoptadipa. Yes, yes, Shoptadipa, you start the session. Welcome the chairperson and start the session.
Yes. Hello. Thank you for the invitation. I'm here. Yes, please.
Yes, yes, I'm here.
Yeah. Good morning. Um, sure, sure, sure. Go ahead.
Uh, should I start the session?
She might be Yes, Dr. Sharma, please begin.
Yes, yes.
And I think I would ask all the speakers to maintain the time. Okay.
Okay. Thank you. A very good afternoon to all of you. I'm Dr. Barnali Sharma, Assistant Professor, Kaliabar College, Assam. The title of my study is a study on traditional practices used for immunity boosting during COVID-19 pandemic situation in Assam. Actually, it was a UG dissertation work of one of my students, Rintu Mani, and she was about to give the presentation, but due to some unavoidable reasons, she couldn't, and I am presenting on behalf of her. We know what is coronavirus. In
Just,
Assam, the first,
could I just could you put your the slides in presentation mode? Then it would be better.
it's there, I think. It's not
No,
coming.
no,
Okay.
uh,
It's there actually. Stop presenting. Okay, okay.
no, just make it full screen.
Okay. Just wait, ma'am. It was there.
Now I have lost it completely.
Just wait, ma'am. Just wait, ma'am. Is it there? Is it coming? Is it coming? Yeah. Okay, okay. The we know the what is actually the COVID-19. Then in Assam, the first case of COVID-19 was identified on 31st March 2020, and from 25th March.
Yeah, please make it full screen. That is what I was telling.
Is it okay? Is it okay now? It's going.
No, it's the same as before.
It's in full screen in my computer actually.
Okay, never mind. Go ahead. Go ahead. Please go ahead.
Okay. Actually, we could have sent all the presentations to the departmental mail ID, isn't it? It would have been better actually. So from 25th March 2020, lockdown was started in Assam, and during this lockdown, people were faced different issues like economic difficulties, and then food insecurities, domestic issues, mental and physical health issues, effect on education, etc. And thousands of people they have lost their life. So uh, as there was no any effective antidotes against coronavirus during the lockdown period, people of Assam started practicing some traditional and home remedies to strengthen their immunity. And according to the World Health Organization, we know what is actually a traditional medicine. Traditional medicine is the sum of knowledge, skill, and practice based on the theories, beliefs, experiences indigenous to different cultures, whether explicable or not, used in maintenance of health as well as in the prevention, diagnosis, improvement of treatment of physical and mental illness. So, the objectives of this study were to gather information about beliefs, knowledge, skills of people of Assam on traditional remedies to boost immunity, which can help to combat against COVID-19, and to collect information regarding home remedies and traditional medicines those people were using actually during the period. So for this, the materials and methods were to study. The study was carried out through an online platform, like using.
Is it that I am having some problem? I don't see any change in the slides. I'm sure because,
Is it?
or is it for
No,
everyone?
actually, I also don't see any change. And I can't see anything. Now
Only
I can
now
see
it has
data.
changed. Only now
But
it has.
it's here. Uh,
Yeah, just now
just do
the
it.
results came, but
But,
earlier it
but
was only the first slide of
But it was
introduction.
going
it's
like
not
that.
visible right now.
Now it is visible.
It's
Yes,
materials and
yes.
methods, yes.
Okay, okay. So a study was carried out through online mode in different regions of Assam. All the participants were of different age groups starting from 16 years, and data were collected by using a questionnaire created on Google Form, and the link was then distributed via different social media platforms. In some cases, data were collected through telephonic conversations. And I'm not giving the questionnaire here. It was there, but it will be due to the time limitations. I'll directly go to the result. Information from 250 respondents from different parts of Assam was collected, of which 55.2% were male and 44.8% were female. 26.4% people believed on medicinal plant. 88.8% showed their dependency on home remedies to cure common health diseases. About 38.8% people were using different kinds of fruits and vegetables as natural food source of vitamin C, such as lemon, guava, amla, banana, tomato, potato, broccoli, orange juice, papaya, pineapple, watermelon, etc. 29.2% people were taking some natural food source of zinc like meat, nut, vegetables, milk, and other dairy products, eggs, cashews, different types of legumes, seeds, whole grains, peas, etc. About 8% were shared home remedies of traditional medicines they have used during COVID-19 pandemic situations, such as doing yoga regularly, eating healthy food, chewing 2 to 3 leaves of tulsi every morning, taking raw turmeric like once tablespoon every morning in empty stomach, then running and jogging and drinking warm water, cinnamon tea, warm water with lemon and honey, turmeric in milk and warm water, karha, warm water with ginger, black pepper tea, etc. 6.4% people shared about traditional ways that their infected family members used to get over from COVID-19 infection, such as taking turmeric in warm water, practicing yoga, and taking vitamin C and zinc rich food, warm water at morning, ginger honey tea, taking healthy food, etc. Some of the graphical representations of the questionnaire, selected questions from the questionnaires I have given here. Like the number 12 question was, do you believe that using traditional medicine can boost your immune system? And about 93.6% of the respondent believe that yes. And do you believe that traditional medicine and home remedies can help in keeping COVID 19 away and minimize the effect of the disease? And of this, most of the people they saw their result as don't know. What are you doing at home to keep your immune system strong against coronavirus? Then lots of people, the categories were like taking antioxidant rich food, taking supplements, practicing yoga, taking vitamin C and zinc rich food sources, etc. And lots of people they have given different people they are giving different um, results, different answers. And what do you drink in morning? The options were warm water, karha tea, ginger honey tea, turmeric in warm water, and others. And about 99 respondents, actually 39.6%, they they showed the result at warm water, and tea was the highest taking beverage. Have you used traditional home remedies to cure common health disease in past days? Then most of the people they said yes. And do you practicing yoga regularly to keep your immune system strong? Then the no answer was actually the negative answer was more than the positive. And here I have given a list. Actually, the it was a recorded list during the question interview and questionnaire session. We have found some name of some plants and their uses, and I have given here the local name, scientific name, and the use part and use medicinal use are given here. The first one is Doron, Leucas, cephalotus. The leaves are used actually, the raw leaves and smoke leaf juice are used and taken with honey. Then hizu, euphorbia antiquorum, here also the leaves are used. Then bahok, it's a kind of bahok tita, it's justicia adatoda, and here also the leaves are used. Then piaz means onion. Tulsi was the highest used plant during the pandemic situation. Then long, dalchini, haldi, jaluk means the piper, and ada means the ginger. And zaifal was also found to be used by Assamese people. Since the time was very less for the survey and it was a simple dissertation work, so um, other work couldn't be done here. And the summary was, since we couldn't do direct personal interview, it was difficult to get views from elderly people through online survey in different places of Assam effectively. Many home remedies known for the survey for keeping COVID-19 away or minimize the effect of the disease by using some ingredients that is easily available at home, which have been previously studied. It's already recorded data were found to be repeated here. And from the survey, we found that traditional practices are mostly common among participants from village area. Maybe traditional medicines are prepared from natural and traditional origin, and village of Assam are endowed with lots of medicinal plants. It is more dominant in village area than in township. They have been using various types of home remedies to maintain health or to treat ailments from past days. And due to less harmful and eco-friendly nature of traditional medicine, villagers of Assam basically depend on that. Since medicinal plants are not easily available in town area, so people from those area are mainly dependent on pharmaceutical products from allopathic medicine. These are some selected references. Thank you.
Yes. Thank you, Dr. Sharma, for your nice presentation. I have one query that it was a questionnaire based survey where what I understood was you've asked people about their faith in traditional medicine. Do you
Not only
huh?
yes, ma'am.
Yeah.
Yes, ma'am.
And what people were given, what type of traditional medicine were being served to those people who were infected with COVID. So, do you have any kind of data on how far it was effective, this traditional medicine, in preventing COVID, and how much it was effective in kind of reducing symptoms of who already got infected with COVID.
Yes, yes, ma'am. That was actually the purpose of the survey. And since
Mm.
it was a simple dissertation work, so even at the letter, the respondents' number was also been increased actually, but we couldn't show it here. But during the survey we found lots of families like they are some of their family members, I have already told here, some of their family members they have already infected with COVID, and due to their habit of taking this type of karha and all, they got means relief, they get rid of the diseases very quickly, and they are that additional like uh, diseases like coughing and all, and they can reduce the effect of the disease actually, and it was proven means they have told that because of this only the risk of the COVID was a bit lesser for us like that.
Yeah. So if you had shown those data, it would make it more interesting.
Oh
Thank
yes,
you.
yes, ma'am. Actually,
Thank you.
it was yeah. Thank you.
Dr. Sharma, I have one question.
Yes, sir.
You use these homemade remedies. So, do these remedies have some side effects as well? So, what is your idea on that? Number one, and number two is, do you have any idea about the mechanism by which these substances they act on COVID?
Okay, actually, the aim was not about the COVID. It was actually to boost the immunity. The aim was the traditional medicines used by common people to boost their immunity so that any kind of viral disease they protect their body from any kind of viral diseases like in during the pandemic period we can see that lots of people we are actually not facing any kind of flu actually during that period, so it was actually the aim that how we the traditional medicines and traditional um, techniques we start and boost our immunity, and the harmful effect I don't think so because people are using and they are not getting any kind of harmful effects like seven plus it's if a person is taking seven plus then maybe the due to the sugar content he or she may get some disease, some other kind of disease, but seven plus is otherwise good and lots of people they are using during this pandemic also.
Okay, thank you.
And this was actually not the aim of this survey.
No, that I
How they are.
understand. That I
Yes,
understand.
yes, yes, sir. Yes, sir.
Yes,
Yes,
I
sir.
just
Actually,
asked you
that
out
would
of
be
my
the.
curiosity because
Yes,
since
yes,
you're
sir.
claiming that these are working, so we should know that how they are working.
Oh, yes, sir.
So I just ask you out of curiosity. I know
Yes, sir.
it's
Thank
not
you.
your aim. Thank you
Yes,
very
sir.
much.
Thank
Thank you. So first I would like to share my screen. Is it visible?
Yes, the only desktop is visible.
Oh yeah, just 1 minute. What?
You can ask
I'm
the next presenter to stay ready, and then so we don't waste.
so I'm sharing, but I don't know why it is not opened.
I don't say that. I mean, ask the next presenter to stay ready so that once they don't waste time in between. We can give him 1 or 2 minutes more here.
Hello.
Yes.
Yes, I can hear you.
Your presentation is not visible.
If there is any problem, can you move out?
Okay,
Yeah,
okay,
now it's
okay.
visible.
Yeah, it is.
It's visible.
Now it is visible, I think.
Yes.
Okay, okay. Okay, so good afternoon to all of you. So sorry for the interruption. Myself, Dr. Biplab Mondal from Department of Zoology, Vidyasagar University, and today I'd like to deliver my presentation that is diversity on HPV family and its causal relation with cervical cancer. So all of you know that cancer it's nothing but it's an uncontrolled proliferation of cells beyond body's homeostasis, and there are some positive agents with the physical, chemical, and biological. And I would like to mention that my emphasis on the biological agents, that is the positive agent HPV, human papilloma virus. And before joining in our university, I worked in the Department of Virology, that CNCI, Chittaranjan National Cancer Institute. And here, my work is on the status of HPV before and after therapy. So, These are the viral cancers, and my emphasis is on the human papillomavirus and their immune cells with the cervical cancers. And I'd like to emphasize on the positive agents and its working mode. And after all, my aim, my objective is to generate awareness among the people regarding this. And these are the types of different types of cancers like oral cancer, breast cancer, pharyngeal cancers, then anal cancer, penile cancer, such and such. And today my talk is on the cervical cancer and step-wise depiction of the most incident cancers in India. I don't like to elaborate this. And it is year-wise, according to the ICMR, year-wise depiction of the cancer prevalence. Now, cervical cancer in India, every year, 132,082 women are diagnosed with cervical cancer and 74,000 die. By 2022, an estimated of 205496 new cases, and it is the death rates due to cervical cancer will occur in India, contributing 30% of global burden, and all women aged 15 to above are at risk of developing it. The most affected age group is 40 to 55 years. Cervical cancer is rare in women in less than 20. Okay, and these are the risk factor for cervical cancer. That the cervical cancer are caused by HPV. Sexually transmitted HPV infections are mostly silent, that are asymptomatic. And other risk factors include having a sex at an early age, multiple sexual partner, women whose mother took diethylstilbestrol during pregnancy or prevent miscarriage, weak immune system, and HIV infections. And the estimated new cervical cancers cases per year, 5 lakhs, of which 79% occur in the developing countries. And cancer cervix occupies either the top rank or the second among the Caesarean women in the developing countries. Now, uh, this is the picture. I would like to mention that the cervix, it is the lower part of the uterus, connects to uterus to the vagina, and these are the part ectocervix, then endocervix, and transformation zone. And this is the part, this is the fallopian tube, then ovarial, it is the portion of the cervix. And for your information, this transformation zone, which connects the ectocervix and endocervix, this region is very much susceptible or prone to HPV infection. This is the another location of the cervix, and this one is the side view of the cervical region. Okay. Now, this is the HPV family. So, human papillomavirus family, there are more than 90 strains are there, and according to their severity of the disease, the positiveness of the disease, these are categorized in two types. First one is the high risk type cancer, and second one is the lowest types of cancer, which mainly causes the what life. And these are the types: low risk type, most common 6 and 11, and this is the genital and oral warts, respiratory papillomatosis in case of child, and low grade cervical cap abnormalities are there. And these are the types only. Most common is the high risk type of cervical cancer, depending upon the severity of the disease, and the causative agent is 16 and 18, and the disease is low and high grade cervical pap abnormalities, cervical cancer, vulvar, vaginal, penile, and the anal cancer, and head and neck cancer also. It's a blueprint genome organization of the HPVs. The genome of HPVs consists of circular double-stranded DNA about 7.9 kb, and the DNA exists in circular form in the viral capsid and replicates as an episome in the nucleus of infected cells. In malignant lesion, it is often found irreversibly integrated into the chromosomal DNA, and it is believed that this insertion could contribute to carcinogenic progression. And it's a brief of the genome, HPV genome, and half of the genome contribute open reading frame. Open reading frame like E6 and E5, E7 protein. In this regard, I would like to mention that there are two kinds of protein, the early proteins, and besides this, late protein are also there. In contrast to early genes, two late genes, that is L1 and L2, are the viral capsid proteins, which accounts for approximately 40% of the genome. 10% of the genome is referred to as the lung control region, and this lung control region doesn't code for proteins. And LCR contains most of the cis responsive elements, that is the replication origin promoters, transcriptional enhancer, and polyadenylation sites. And according to the Harold Joe Husserl, is the pioneer of this HPV discovery, and this is in brief the life cycle of the human papillomavirus infection. It requires the availability of the basal layer cells, and they usually occurs in the microlesions of the skin. And the infections in infected cell divides the population spread laterally. Some of the progeny migrate into the suprabasal differentiated cells, and where viral genes are activated. So this is the brief life cycle or the spreading capability of the HPV gene. And now, what is cervical cancer? Cervical cancer is a condition in which the cells in the lining of the cervix changing and grow very fast, producing a mass of the cells called tumor. And another route of HPV infections, that is genital contact, vaginal or anal sex, transmit to newborn from infected mother during vaginal delivery, and child may develop warts in the throat, that is RRP, recurrent respiratory papillomatosis. 80% of the cervical cancer causes due to high risk. Earlier, I already mentioned that there are two kinds of high risk and low risk. So HPV 16 and HPV 18 are the high risk type of cervical cancer, and most of the cancer, that is 80% cervical cancer, is Caucasian. Now, the genital warts, I mean, the responsible element is the HPV 6 and 11. These are the low risk type of HPV status, and it may cause genital warts or condylomata acuminata, that is the vulvar, urethra, cervix, anus, penis, and scrotum region may cause. And how do genital wart look like? So genital wart don't always look the same, usually soft, less colored growth, but they can be raised, so flat, small, or large, and alone or in cluster condition. And depending on the severity or the invasiveness of the particular disease, it may be categorized in CIN1, CIN2, and CIN3. That is, first one is the mild type, moderate type, and severe type of intraepithelial neoplasia that it may develop depending on the severity. Now it is again in the cervical CIN1, that is mild dysplasia includes condylomata, then moderate dysplasia, and in case of several severe dysplasia, that is CIN3. So now this is cancer, cervical cancer biology. If we go to this slide, that normal cervix, when got the HPV infections, then it may cause persistent HPV infections. So normal to persistent HPV infections, and after that cervical dysplasia, that is pre-cancer to cervical cancer. So this is the gradation of the. Okay, okay. Thank you. So just I need only 2 minutes. So this is the picture, the normal to CIN, and normal cell to cancerous cells. These are the differences. How the nuclear pattern changes in two differences. And Pap smear is the one of the important diagnostic tools. According to the George Papanicolaou, in the year 1940, he discovered that Pap smear is the only diagnostic method for the discrimination of that cancerous cell from the normal one. So this is the first. And these are about the staging of the cancer after diagnosis, the stage 0, stage 1, stage 2, and after that, according to the American Cancer Society, it is established that the different gradation of the cervical cancers and the sites of cancer progressions. And now the prevalence of HPV, the 20 million Americans are currently infected with HPV, and the American Cancer Society estimates that 11070 women diagnosed with cervical cancers. And these are the detection of cervical cancer: colposcopy, biopsy, then Pap test, PCR technique, southern blotting hybridization, immunohistochemistry, and hybrid capture assay. Due to the scarcity of time, I cannot elaborate these techniques, but these are the, and so far as the treatment is concerned, that surgery, chemotherapy, and radiation therapy. Gardasil is the vaccination method, vaccination, and another one is the Cervarix. So starting from the day 1, then month 2, then month 6, and after vaccination, follow up follow up with a pap test, and it can reduce the cancer burden in gardasil and cervix. So these are the side effects of the vaccines. So redness, headache, fever, and if you become pregnant, so there may be risk of the newborn. Now, we should not receive the vaccines. These are the persons, and 7 warning signals of the cancer. First one is the change in the usual bowel habit. A sore throat that doesn't heal, unusual and repeated bleeding, thickening of the lump or any part of the body, indigestion or difficulty, then obvious change in the size and color of mole, the nagging cough and persistent worsening. So these are the 7 warning signals of cancer, that is CAUTI. So these are the doorsteps of cancer, that is ignorance, illiteracy, shyness, and fear, scarcity. So we are swimming in a sea of carcinogen. We should be alert by early checkup, regular screening. We should spread awareness among the people to organize camp for early detection of cancer, because effective measure of cancer control is the early detection. So these are the bibliography for preparing this slides. I tell, and it's online resources. And these are the publications we have made during our work. First one, second one, and third one. And the person, Harald Jorgensen, I mentioned that he is a German scientist in the field of virology, and he got Nobel Prize in the year 2008 for known his discovery that HPV can cause cervical cancer. And I believe that this chronic
Good.
disease demands
I
persistent
think,
involvement from the scientific side.
Dr. Mundal, we have exceeded our time a
Okay, okay,
lot.
okay. Thank
Yeah.
you. So,
So
okay,
I have a question for you.
yeah.
It would be. It was a nice presentation, but I had a question.
Yeah.
Could you highlight on? It would be better if you would highlight on what are the oncoproteins from the human papilloma virus that are responsible for the transformation.
Yeah, ma'am.
Right? And what are they associated with? What is the cause that this gives rise to?
Actually, thank you, ma'am.
Because the last slides, they were, few slides that you showed, they are quite general for any type of cancer.
Yes.
Not just related to cervical cancer.
Yeah, actually, ma'am, for your query, that is early proteins, that is E6 and E7 protein. These are the very vital for causation for causative agent, and we know that P53 and RB genes these are the tumor suppressive gene, and E6 protein and E7 protein are the responsible for cervical cancer. Yes, and P53 protein, it can suppress the activity of the A6, and RB can suppress the activity of the E7 protein.
Yes.
Thank you.
So more depth into about the human cervical cancer related papilloma virus would be more informative. I think.
Thank you.
Thank you. I have no other questions. If there are questions from
no other questions from me.
Yes, ma'am. Is it visible, ma'am? So, good afternoon, everyone. My name is Golok Mujumdar, PhD research scholar from University of Gorbongo under the guidance of Dr. Shyamapad Mondal. Today, I am going to present my paper entitled Bioinformatics Approaches to Explore the Inhibition of 3CL-Pro of SARS-CoV-2 by Phytochemical Ligands. So first, background. The ongoing COVID-19 coronavirus disease with SARS-CoV-2 severe acute respiratory virus infection is now a huge global health crisis. In absence of an effective treatment of COVID-19 and in view of the adverse toxic effect of currently available drugs, the phytochemicals are compared to safe and effective against diseases. So develop a potential drug against such conditions. In silico method is very much explorative to repurposing some drugs against COVID-19, like L-hyoscamine from Atropa belladonna, Eupatorium from Eupatorium parfoliatum, and alkaloid L-toltecan from Lycopodium. So our aims of the study is to retrieve these bioactive compounds and perform molecular docking, molecular simulation against SARS-CoV-2 target protein 3CL-Pro, because 3CL-Pro is an excellent druggable target because its crucial role in coronavirus polyprotein into a functional component for that is vital for viral replication, and also to determine the following phytochemical ligands, pharmacokinetic properties, bioavailability, and toxicity levels. Here is the methods. For this study, we use the following methods. Like for retrieval and preparation of ligands, we use PubChem database and UCSF Chimera. For retrieval of protein and preparation, we use RCSB Protein Data Bank and UCSF Chimera. For drug likeness analysis, we use PKCSM web server. For molecular docking, we use UCSF Chimera. Ramachandran plot was generated using Procheck software. Molecular docking were done using GROMAX version 2021, and binding energy was calculated by MMPBSA approach, and visualization were done using DMD and Discovery Studio Visualizer. And here is our target protein, crystal structure of SARS-CoV-2. The main protease and retrieved from protein data bank. Here is the protein data bank ID 7K3T. And here is the Ramachandran plot of the protein 3CL-Pro. And the residue in most favored region in this plot, we found that residue of most favored region are in Ramachandran plot is 90.2%. That means we can say that this protein is a good protein for our study. And here is the selected ligands like L27 alkaloid, L-hyoscamine, and eupatridium.
It's not visible, Golok. It's not visible.
Ma'am, is it visible, ma'am?
It's visible to me.
I can see. Yeah, it's visible. You can carry on.
Okay, sir. Okay, sir. Here is the selected ligands L27 alkaloid, L-hyoscamine, and eupatrium with their ATP name and with PubChem ID. Results. So after molecular docking, the three ligands were docked within the pocket created through a pocket. Here is the ligand molecule. Red one is belladonna, yellow one is eupatrium, or magenta one is lycopodium, with the binding energy minus 6.9, minus 6.5, and minus 6.8 kilocalorie per mole respectively. And the minus 6.5 equals or less than minus 6.5 kilocalorie per mole were considered as a good binding results for the analysis. And here is the docked complex 3D and 2D diagram are shown. And the 2D diagram, the 3CL-Pro and belladonna it shows that the three conventional hydrogen bond GLN 107, GLN 110, proline 108, and the carbon hydrogen bond were formed with proline 108 and glycine 109. Here is the lycopodium. The bond, three conventional hydrogen bond were formed with GLN 110, histidine 246, and glutamine 107, and the carbon hydrogen bond with proline 108. And here is the third ligand, the eupatridium, with two conventional hydrogen bond with histidine 246 and carbon hydrogen bond with glutamine 109, proline 132, and glutamic acid 240. So next is the molecular dynamic simulation. To analyze this, the stability and dynamics of APO 3CL-Pro without ligand and holoenzyme 3CL-Pro with ligand, simulation carried out for 100 picosecond using GROMAX version 2021 with charm force field. Here is the solvated complex and the key hydrogen bond interaction. The AB is the belladonna, and C and D is the eupatridium, and E and F is the lycopodium. So during the simulation, 300 Kelvin temperature attained based on the Maxwell distribution, pressure 1 bar, and it is fluctuated during equilibration. Evidence suggests that evident from the large RMSD. And here is the RMSD of backbone after square after least square fitting to backbone atoms range from 0.12 nanometer, implying stability of the structure. And in this case, distance between backbone atom is 0.09 to 0.10, and ligand heavy atom were 0.046 nanometer for eupatorium, 0.056 for hyoscine, and 0.011 for belladonna. So the position distance applied to both apo and hollow protein in our study and did not impose any variation in the crystal and equilibrated structure for both RMSD and RMSF of backbone atoms after least square feet to the backbone and ligand heavy atoms. So here is the simulated complex of 3Cl Pro belladonna with net binding energy minus 78.96 kilojoule per mole. Here is the 3Cl Pro and lycopodium simulation structure after the net. We get the net binding energy minus 114.17 kilojoule per mole. And here is the third ligand, last ligand, eupatorium with binding energy, net binding energy minus 1 to 1.3 kilojoule per mole. Admit analysis and drug likeness. The bioavailability radar values are lying within the pink zone with bioavailability score 0.55. And in case of eupatorium, saturation value was slightly lower in terms of infraction CSP. And here is the three ligands with the Lipinski rules of 5 parameters: molecular weight, log P, and H bond acceptor and H bond donor. And there is no violation of Lipinski rules of 5, indicating their acceptability for oral administration. Here is the ADMET profiles of three ligand molecules, and the in silico toxicity by ADMET showed shown that none of the ligands possessed hepatotoxicity and cytotoxicity except eupatorium. AMS test is positive. Next is the conclusion. So based on upon the net binding energy suggesting that all the ligands molecules achieve thermodynamically stable complex with SARS-CoV-2 3Cl Pro, ADMET profile bioavailability score indicated their suitability as a potential drug candidate. So the current study is authenticated by MD simulation, docking analysis, and the inhibitive property of bioactive antiviral phytocompounds belladonna, eupatorium, alkaloid L27 from lycopodium due to their good binding and energetically stable complex formation with 3Cl Pro of SARS-CoV-2. It indicates that it can be used as a potential drugs against COVID-19. And the acknowledgement, I wish to extend my deepest sense of gratitude and special regards to my PhD guide, Dr. Shamapada Mondol, Dean, Faculty of Council for Education in Citizen Science, and my special thanks to International Conference on Integrated Approach Towards Sustainable Management of Environment for Safe and Food Nutrition, Improved Health, Organizing Committee for their excellent support. And here is this work is published in drug discovery journals. So thank you.
I have a few questions to Golo.
Yes.
My first question is, you showed the RMSD graph, but it's not very stable. There are lots of fluctuations. So how do you consider that graph to be the plateau is reached? See, there are fluctuations.
Yes, ma'am.
It has to be linear.
Yes, but we just use 100 picosecond study.
No, but why did you choose 100 picosecond? You could have extended.
But ma'am, our operating system is not suitable for that, so we use only for 100 picosecond.
From this graph, I don't think it is very conclusive that the drug is interacting properly, because if there are fluctuations, in that case, the interactions change.
No, the binding energy also I get is good for molecular docking binding energy.
No, is the binding, and did you check the binding energy throughout the trajectory?
Molecular docking and after simulation we
no, I'm not talking about the final binding energy. I'm talking about the stability. Like when you are saying that this drug is interacting, so the binding energy has to be like in a range. So at that range, like you said 6.4 or 6.2, something like that. That particular range has to be maintained throughout the trajectory.
Okay.
So did you check that? And one more thing that I'd like to mention is that in some cases you used kilocalorie and in some cases kilojoules. So it's better to use just one particular unit.
Okay, ma'am.
I don't have any other things.
Thank you.
I would just, overall, it was a nice presentation. You showed some of your results. So,
Yes, ma'am. Have you any question, ma'am? All right.
The overall presentation was very nice. So Golok, you just write down or take the whatever the Dr. Bakchi has said, the suggestions and the whatever
Yes,
the
sir.
queries. So you can work on that so that your work would be better and you can publish in a very good paper. Okay, thank you.
Okay, sir. So this paper already published, but I am working also this field on.
Okay, okay. So maybe there may be
Yes,
other
sir.
papers that may be coming out, so
Okay,
you can
okay,
take down all these suggestions. Okay.
okay, yes, okay, sir.
Yes, yes, ma'am. Am I audible and my slide is visible? Okay. So good afternoon, everybody. This is my privilege that my abstract has been accepted for the presentation in your university. I am Dr. Srikanto Guria, Assistant Professor, Postgraduate Department of Zoology, Barasat Government College, Kolkata. My topic is peritoneal macrophage at the crossroads of diabetes mellitus and thyroid dysfunction, a short report. What is the aims and objective of the topic? I have done this work with my supervisor, Professor Madhusudan Das, former head and dean of science, Department of Zoology, University of Calcutta. The aims and objective is that study of cytomorphology of hypo and hyperthyroid macrophage in rat, and the death nature of the hypo and hyperthyroid rat peritoneal macrophage, whether it is apoptosis or necrosis, study of phagocytosis of rat peritoneal macrophage, and study of phagocytosis of rat peritoneal macrophage is mainly with the charcoal particle and the yeast particle cell viability assay, and whether then any effect of macrophage mediated immunity on thyroid dysfunction or not. So what we have done, we treated the rats with the methimazole for hypothyroidism, and we have treated the rats with thyroxine or L-troxine for hyperthyroidism, and also activated charcoal particles suspended in the vertebrate saline or 0.1 mole phosphate buffer saline and injected to the rat peritoneum. Then macrophage was isolated or extracted, fixed with methanol, stained with Giemsa or also trypan blue for cell viability assay, and counting of the macrophage, and so define the result. So the serum T3 and T4 was reduced in hypothyroid rat, whereas it is significantly elevated in hyperthyroid rat as expected. So this is the picture of the Giemsa staining of the macrophages, where this is the macrophage, and the different types of the phagocytosis was found in case of the control macrophage. This is the macrophage, and these are the activated charcoal particles. The macrophage were engulfed the activated carbon particle or charcoal particles, the surface adherence or surface binding of the charcoal particle on the membrane of the macrophage. But cytomorphology of peritoneal macrophage in the hypothyroid group was altered. Pycnotic and necrotic macrophage in the hypothyroid group were not able to phagocytose the charcoal particles. The right-hand side picture, it was found that apoptotic macrophage and most of the charcoal particles are present outside the cell. It means the cells were not able to phagocytose the activated charcoal particle. Then we have also studied the macrophages by staining with eosin and hematoxylin and fluorescence microscopic analysis. In case of the normal rat peritoneal macrophage, the left hand side, it is found that eosin hematoxylin saying the normal macrophage, the membrane is intact. But in case of the right hand side, it is found that in hypothyroid rat peritoneal macrophage, the membrane bleeding was found. And in below the left hand side, it was found the intact membrane of the nucleus when stained with the DAPI, and the right hand side, the nuclear fragmentation was noticed. Actually, what is happening, thyroid hormones are essential for normal function of the macrophage. Their effect may be direct or indirect as per Liu et al 1993 or Rosa et al 1995. Now, the macrophage counting, it is also reduced in case of the hypothyroid rat. So this study may clearly indicate the hypothyroidism adversely affect the peritoneal macrophage function. Then phagocytosis of yeast particle by peritoneal macrophage in normal and hyperthyroid rat. So the upper pictures, that is the normal macrophage taking the yeast particle. The yeast particle were stained with the eosin. It is found that the yeast particles were adhere on the surface of the macrophage, and then one by one the macrophage took the yeast particle by the process of the phagocytosis. And this is the higher magnification picture where found many yeast particle adhere on the surface of the macrophages, and two macrophages were taken the yeast particle or engulfing one yeast particle. But in case of the hyperthyroid rat, most of the yeast particle present outside the cell because the macrophages become necrotic and membrane bleeding also found. So hyperthyroidism altered rate of phagocytosis of yeast by macrophages. We have also calculated the phagocytic index, and the phagocytic index, it was found that the hyperthyroidism, the phagocytic index were significantly decreased. So what is the types of the death, whether it is apoptosis or necrosis? But when we analyze the type of the death, it was found some of the cells showed peritoneal macrophage bleeding, it very much similar to the apoptosis, and some of the cells showed the membrane rupture that released the cytoplasmic content that was similar to necrosis. So both apoptosis and necrosis found in the slide. So we have also published this result in a paper. Now I came to the diabetes. We treated rat with the streptozotocin and also alloxan. So what is the result? Rats were divided into experimental and their respective control group. The experimental rat were treated with the streptozotocin, and actually before going to the result, I can explain the mode of action of the streptozotocin and alloxan. Both are glucose analog, and both can enter to the pancreatic beta cell by GLUT2, that is glucose transporter 2. And the difference between the streptozotocin and alloxan is that streptozotocin promote the DNA alkylation and promote the apoptosis or DNA breakdown of the beta cell, and alloxan produce the reactive oxygen species that can that can lead to the death of the beta cell. So both are responsible for the diabetes. Now, what is the observation? The right hand side upper picture showed that these are the normal macrophage, but the arrow-headed cells, it is the pycnotic and the necrotic macrophage. Significant amount of the peritoneal macrophage become pycnotic or necrotic, indicated by the arrow. And also the necrotic macrophage were not able to take the charcoal particles. It is the lower handed left-handed side picture, that is the necrotic cells, and most of the significant amount of the charcoal particle present outside the cell. But few macrophage, it was striking that they were very much loaded with the charcoal particles. This is the picture, that is the right hand side lower picture, the charcoal particle laden macrophages. Now, this is the mean phagocytic index in normal and diabetic group. Automatically, the diabetic macrophage that is treated with the streptozotocin showed very reduced phagocytic index. There are majority two types of the macrophage we found. One is the LPM, that is the large phagocyte, large peritoneal macrophage. Another is type is SPM, small peritoneal macrophage. Two events were found, that is the fusogenic activity. Small peritoneal macrophage can fuse with the large peritoneal macrophage. It is number one. And number two, the aggregation property of the macrophage. Both the small peritoneal macrophage and large peritoneal macrophage, they showed the aggregation. That is, increased cell aggregation as well as increased tendency of the macrophage fusion was noticed in the rat when treated with the streptozotocin. Another chemical, that is the alloxan, that can also promote the rat become diabetic. In case of the upper hand left hand side picture, the peritoneal macrophage intact nucleus and membrane. Okay. And but in case of the right hand side, the necrotic macrophage also found, and it is the most of the dead cell showed trypan group positive response. So what is the inference or conclusion? The conclusion is that the diabetic macrophage may be related with the inflammatory reaction. Most of the macrophage are classically activated macrophage rather than alternatively activated macrophage. And classically activated macrophage are associated with the inflammatory reaction. In case of the hypothyroidism, mainly the methimazole is responsible for the reactive oxygen species that promote apoptosis of the macrophage. So we have published this paper in recent Advances Animal Science Research. Then we have also published, this is my publication along with Professor Madhusudan Das. Recently, I have published that streptozotocin induced diabetes mellitus promote alternation of peritoneal macrophage morphology and behavior. Thank you for attention.
Thank you, Dr. Guria, for your very nice presentation. You mentioned that the number of macrophages decreased in case of hypothyroidism. What is the case in case of hyperthyroidism? Does it also affect the number of macrophages? And in case of diabetes too, is there any influence on the number?
Yes, we counted the macrophage in both hypothyroidism and hyperthyroidism. The macrophage number, the peritoneal macrophage number is decreased when we have counted in the hemocytometer. And in diabetic case also, the peritoneal macrophage number decreased. But if you analyze the tissue, if you analyze the pancreatic tissue, in case of the diabetic, it is found that some macrophage increased in number. Actually, they are the classically activated macrophages that are related with the inflammation.
Okay. Thank you. No other question? If somebody else wants to ask.
I have no other question.
Okay, Dr. Guria, you have a very nice presentation. So I have just one query. You used only 600 micromolar thyroxine. So have you standardized that dose, or it's from previous paper that you selected that dose?
Yes, sir. Actually, it is from the previous paper, because the previous studies they have the result in that dose and published the paper. So I took this dose.
So you didn't study other doses like 400 or maybe higher than 600. So you just only studied the 600 micromolar.
Yes, 600 micromolar. I have just studied that for L-troxin or thyroxine.
Okay. Best of luck. Okay.
Thank you, sir.
Thank you.
Yes, ma'am. Good afternoon, everyone. I am Rinmay Sengupta, and I'm a PhD scholar of Gaurbanga University, and I am under the guidance of Dr. Mitali Das of Gaurbanga University. She is the assistant professor of Department of Zoology. My topic is dietary and lifestyle management of osteoporosis under the theme health and disease. Osteoporosis occurs when bone lose their strength and thickness due to a loss of minerals that the body has difficulty in replacing it. And who commented on osteoporosis that it is not only the major cause of fracture, but it ranks among the highest disease that causes people to become bedridden with serious complication. And also, IOF, that is International Osteoporosis Foundation, projects that 50% of the osteoporotic hip fracture will occur in Asia by the year 2050. There are several causes of osteoporosis. Mostly, it includes low calcium intake, vitamin D deficiency, low bone mineral density, smoking, alcohol abuse, increasing age, female sex, post-menopausal status. So, among these, calcium deficiency, vitamin D deficiency, and alcohol abuse, also smoking, this will be further elaborated. We have seen that female gender is more prone to osteoporosis. It may account that in this incidence of osteoporosis, as women have a lower peak bone mass, and partly because of the hormonal changes that occur at the menopause. Incidence of osteoporosis says that globally more than 8.9 million fractures are caused by osteoporosis per year, and currently it is estimated that 200 million people are suffering from osteoporosis. Recent studies says that according to International Osteoporosis Foundation, 1 in 3 women over the age of 50 and 1 in 5 men will experience osteoporotic fracture in their lifetime. There is, as we have seen, that calcium plays a great role in bone mineralization. So approximately 99% of the body calcium is found in bone, and osteoblasts are responsible for new bone formation. There is CASR gene that provides instruction for making a protein called calcium sensing receptor, that is CASR. This calcium molecule attached to the CASR protein, which allows the protein to monitor and regulate the amount of calcium in the blood. So this calcium, it also work in accordance with PTH and acts as to defend against hypocalcemia. This CASR controls the parathyroid hormone and secretion, which in turn, by direct and indirect actions on kidney, and maintains the normal extracellular calcium concentration in the body. This CASR plays a key role in maintaining the homeostasis of calcium in the body. And it is being observed that any mutation in the CASR gene causes familiar hypercalcemia, and which may further lead to osteoporosis. And also we have seen that vitamin D plays great role, and it can be ingested as vitamin D2 and D3 from plants and animals respectively. And vitamin D stimulates calcium absorption. Vitamin D and parathyroid hormone work in concert to mobilize calcium from the bone and conserve the calcium from urine. The biological action of vitamin D exerted through the nuclear vitamin D receptor VDR mediated control of gene. Here you can, this has been shown that vitamin D acts in concert with parathyroid hormone and conserve the our serum calcium level. So the hormonal form of vitamin D, it acts as a ligand for vitamin D receptor, and it activates target gene. And this vitamin D has been shown to affect the rank L expression. This rank L expression will be elaborated further. Research shows that any polymorphism in this VDR had caused lumbar spine, and ultimately it is very account for the osteoporosis. Now, the effect of alcohol on bone metabolism. Alcohol consumption imparts the absorption of calcium and vitamin D. Alcohol can decrease the absorption of calcium via intestine, and it can affect on pancreas and vitamin D metabolism. And this ethanol ultimately shown to disrupt the calcium homeostasis and is known to have deleterious effect on bone. And also nicotine also affect in the bone metabolism. evidence also shown that nicotine, tobacco, this smoke contains more than 7,000 chemical compound and affect bone mineral density directly or indirectly on the rank L OPG pathway, and it hampered the calcium balance in the body and ultimately lead to osteoporosis. This rank L, rank K OPG system, this system regulates the osteoclast formation. Here it has been shown that this rank L and rank K, it stimulates the osteoclast, which is effective in bone reabsorption. But this OPG, this protein binds to the rank L and it prevents the formation of osteoclast, which ultimately prevents the formation of bone reabsorption. But if OPG amount is more, then the bone formation will be more. So this pathway is very vital for the bone, healthy bone, and thereby prevent osteoporosis if the amount of OPG is more. So these were the some uh, genetic pathway accounting for osteoporosis. For preventing osteoporosis, we must take care of the diet. The dietary consumption of calcium and vitamin D is very vital. Calcium and vitamin D rich food includes cheese, green leafy vegetables, and vitamin D rich food includes fish oil and mollusk, oyster, etc. And exercise also account for healthy bone and prevent osteoporosis. This exercise has an impact on this rank L and rank K pathway. There are certain medication reclass Evista, calcitonin, and also hormone therapy and Forteo. This uh, provide a good bone health and preventing osteoporosis. So these are the reference I have included, and I am very grateful to my ma'am who have guided me, and also the organizing committee of this conference. Thank you everyone for bearing me. That's it. Thank you.
Okay, Mrinmoy, nice presentation. Thank you for your presentation. So I have just one query. Have you studied any gene expression, or just you have studied the application of different dietary supplement and their role, how to control this osteoporosis? Have you studied any gene expression? You have mentioned so many pathways and genes.
Sir, kindly mute one's audio because both audio is unmuted. Hello.
I would request Mr. Inamul just yes. Okay, now I can hear. Okay, carry on. Minmoy?
Yes, sir.
Yeah. So my
Yes.
question was, have you studied or did you study any expression of these, the genes, what you have just mentioned, and these are related to your osteoporosis? So have you studied any pathways or gene expression?
Yes, sir. I am working on LRP5 polymorphism, but I have not mentioned here because I have not started working in the lab. So I just did review kind of work. I have just started my work in this year.
Okay. Okay, thank you.
Yes, sir. Thank you.
So Dr. Bakchi, you have any query?
No, I don't have any question.
Okay, Rita ma'am?
No, it's okay. It's okay.
Okay. So we can move on, and thank you, Mrinmoy, for your presentation. So we can call the next speaker. I think it's Nilanjana Bhattacharya.
Yes, sir. Yes, sir. Shall I start, sir?
Yes, please.
Okay, just a minute. Is it visible, sir?
Yes, yes.
Okay, okay. Thank you, sir. Good afternoon, everyone. Myself, Dr. Nilanjana Bhattacharya. Assistant Professor, Department of Biotechnology, Swami Vivekananda Institute of Modern Science, Kolkata. And today my topic is mucormycosis in COVID patients and opportunist infection causes another threat to pandemic. So, in the pandemic situation, we all are facing the pandemic situation. Pandemic COVID-19 continues to be a significant problem worldwide. And while several treatment options have been evaluated, but only glucocorticoids have been shown to improve survival in COVID-19. Unfortunately, the widespread use of glucocorticoids can lead to secondary bacterial or fungal infection. And mucormycosis, we can also say zygomycosis or phycomycosis, is a rapid progressing opportunistic invasive fungal infection predominantly seen in immunocompromised individuals with impaired neutrophil response. In India, during second wave of pandemic, several cases of mucormycosis in people with COVID-19 have been increasingly reported. The primary reasons that appear to be facilitating mucorales spores to germinate in people with COVID-19 are mainly the ideal environment of low oxygen, that is hypoxia, high glucose level, diabetes or hyperglycemia or steroid-induced hyperglycemia, acidic medium, that is metabolic acidosis or diabetic ketoacidosis, high iron levels, and decreased phagocytic activity of white blood cells due to immunosuppression. And the cause of immunosuppression may be SARS-CoV-2 mediated immunosuppression or steroid mediated or any kind of background comorbidity. So we all know that steroids can suppress the cytokine storm associated with COVID-19, and they also serve to reduce overall immune system activity. So as well as pushing up blood sugar level. So several other risk factors are also there. For example, prolonged hospitalization with or without mechanical ventilators. Mucormycosis is not a new disease. It was always reported from people who are immunocompromised, like people after chemotherapy, people with HIV infection, people with high diabetes, like that. And the mucormycosis is also sometimes often referred as the black funk. Basically, this is a misnomer. It is so called because it causes tissue necrosis. Any necrosed decomposed tissue produces hessure, and that is colored black. And as per I have collected the information that in India by late June 2021, more or less 40,845 mucormycosis cases had been reported, most of which affected the nasal passages, brain, and spinal. So next, pathophysiology. Fungi of the order Mucorales are causes of mucormycosis. It is a life-threatening fungal infection, and species belonging to the family Mucoraceae are isolated more frequently from patients with mucormycosis than any other family. And among the Mucoraceae, Rhizopus oryzae is the we can say most common cause of infection. It most commonly affects the sinuses or the lungs after inhaling fungal spores from the air. It can also occur on the skin after a cut, burn, or any type of skin injury. So these infections are usually acquired when spores from the molds either inhaled or less commonly enter the body through a cut or injury that I have mentioned. Mucormycosis develops exclusively in immunocompromised patients who lack the normal defense mechanism. So neutropenic patients are at increased risk of developing mucormycosis. Furthermore, patients with dysfunctional phagocytes are also at high risk. Hyperglycemia, acidosis are inactivate the ability of phagocytes to move toward and kill the organism. So these are the basic pathology. Next, role of iron in mucormycosis pathogen. So iron is an important component for the growth of mucorales in host. And in immunocompromised host, iron is released from sequestering protein and helps to grow fungi within the body. And in case of mammals also, but they are having little bit iron in the serum iron in the form of serum iron, and they are basically available to microorganisms because most of them they are highly bound to carrier proteins such as transferrin. Acidotic conditions disrupt the capacity of transferrin to bind iron, and patients who receive dialysis mainly they are treated with deferoxamine. So this is the chelating iron chelating agent, deferoxamine.
Okay, some background sound is coming.
Hello.
Some background sound is coming. Please switch off. I don't know. It's something. Why it's coming?
Yes, yes. Now it is okay, sir. Thank you. So deferoxamine, a bacterial siderophore, basically, it makes the patient susceptible to rhizopus infection. And the patients with diabetic ketoacidosis, they are high risk of developing rhinocerebral mucormycosis as low pH conditions. So obviously, acidic conditions means low pH conditions, and in that particular case, the conditions cause proton-mediated displacement of ferric from serum carrier molecules. Basically, it helps to convert ferric ion to ferrous ion. That's simple conversion. So we all know that iron is present in two readily available ionization states, ferrous and ferric. So ferric ion is then reduced at the cell surface to ferrous ion. And deferoxamine, in contrast to that acidic condition, it directly chelates iron from transferrin, resulting in iron deferoxamine complex. And this complex is also known as ferroxamine. So ferroxamine then binds to unidentified receptors on the surface of the zygomycetes, and the fungus then liberates the iron from ferroxamine. So ferrous ion is then reoxidized back to ferric ion by copper oxidase. So copper oxidase, it is present in the cell membrane. So this is the basic pathway by which iron initiates the suitable environment for the mucormycosis infection. So next, mucormycosis as post-COVID illness. So due to associated comorbidities, so comorbidities are mainly diabetes, chronic pulmonary disease, and immunocompromised conditions. The patients, they are prone to develop severe opportunistic infections. And among them, the few are, for example, oropharyngeal candidiasis. So it is an infection caused by yeast candida. Candida normally lives on the skin and inside our body, such as mouth, throat, gut, vagina, without causing any problem. But sometimes candida can multiply and cause infection if the environment inside the mouth or inside our body changes in such a way that encourages fungal growth. Another disease, pneumocystic xerophagic pneumonia. This is a fungal infection of lung. Mostly the weakened immune system, the individuals, those they are having weakened immune system, they are suffering from that particular disease. And the disease used to be called pneumocystis carinii or PCP pneumonia. Next, pulmonary aspergillosis. It is basically the term refer to a number of conditions, we can say, and caused by a particular fungus, that is aspergillus. So other than these, rhinocerebral disease may manifest as unilateral retroorbital headache, facial pain, numbness, fever, hyposmia, and nasal congestion. So like that. So these are the basic illness. So this is the diagram showing the different diseases starting from the pulmonary aspergillosis and other diseases and other symptoms caused by mucormycosis. So next, the symptoms of mucormycosis in post-COVID patients. The very common symptoms, if the mucormycosis is related to sinus and brain infection, that means rhinocerebral mucormycosis, then it includes facial swelling, headache, sinus congestion, black lessons, nasal breathe, fever, like that. And if it is related to pulmonary infection, lung infection, then the common symptoms are fever, cough, chest pain, shortness of breath, like that. So these are the most common symptoms of mucormycosis in post-COVID patients. And sometimes what happens, pneumonia caused by mucormycosis and COVID pneumonia, they are very close, and it is very much challenging to determine which one is the COVID pneumonia and which one is the mucormycosis induced pneumonia. So next, diagnosis and treatment. So histopathological examination and imaging method, these two are the most common way by which we can diagnose. The analysis of cerebrospinal fluid analysis, surgical specimen analysis, CT scan, along with that MRI. These are the helpful methods. But other than these, another imaging technique, which is not yet widely used. This is positron emission topography, computed tomography. So this is also, that particular method can help to detect mucormycosis. And the treatment part, so it is almost important to control diabetes, reduce steroid use, discontinue immunomodulating drugs, and to maintain adequate systemic hydration. And treatment basically mainly includes the infusion of normal saline and one drug that is very useful, that is amphotericin B15 per milligram per kg body weight per day, and antifungal therapy for at least 4 to 6 weeks. And the last but not the least, that management of COVID patients with mucormycosis is basically a team effort involving microbiologists, internal medicine specialists, intensive neurologists, ENT specialists, ophthalmologists, dentists, surgeons, and others. Thank you.
Thank you, Dr. Nath, for your nice presentation. So the session is open for discussion, but before that, I have just a little query. It's not a query. You can say there is one antifungal drug that is called the amphotericin B. So is there any report that it has some role in controlling the mucormycosis? Do you have any information?
No, sir. Actually, this is basically an article, and I don't have that kind of statistical data regarding the use and the effect of amphotericin B15.
So this is not widely used drug in case of, or it has not
Yes.
been
Okay, sir.
studied.
So I remember that particular point, and obviously I'm trying to add that part in my article.
Okay.
Thank you.
Okay. So any other query?
Thank you, Dr. Bhattacharya, for a very nice and informative talk. I have just one query. Is there what one should do in COVID patients? Are there any preventive measures so that one does not develop this kind of infection?
Ma'am, pardon, ma'am. Please repeat, ma'am. I can't hear you
My
properly.
question was, are there any preventive?
Can't hear you, ma'am.
Ma'am is asking whether there is any preventive measure that we can take just to, avoid mucormycosis, or I think Madam is asking that question.
Okay. Actually, the best option is to maintain the hygiene, I think. Other than the educational part, or other than the diagnosed part, that is as I have mentioned, that the immunocompromised patients with diabetes, patients with any kind of COPD, they are in the great risk. But other than this, I think hygiene is the best option in our normal daily life, to maintain the proper hygiene. That is the main part, and the rest is the more or less the same, the maintaining the other part.
Okay, thank you.
Okay, Dr. Bakshi.
Thank you, ma'am.
Okay, am I audible, ma'am?
Yes.
Yes, ma'am. I am here. Just
Rajab, you
confirmation.
start presenting. Huh?
Okay.
We can hear you.
Is it visible?
Yes.
Okay. Good afternoon, everyone. Myself, Rajesh Sarkar, PhD scholar from University of Gurgaon. And this topic is related to association of soluble HLA-G isoform with 14 base pair insertion deletion and 3142 GC polymorphism in rheumatoid arthritis. First of all, in introduction, we have a basic idea about the rheumatic disease like rheumatoid arthritis. It is a chronic and autoimmune inflammatory arthritis which affect mainly our small joints of peripheral parts of our body. And there are several factors which is directly as well as indirectly related to the pathogenesis of this disease, like several environmental agents as well as genetic self-antigen like HLA, human leukocyte antigen. And as this is a autoimmune disease, basically it is more common in women compared than the men. And worldwide epidemiological status is like that. It is approximately 0.5 to 1% people affected from the world. There are basic two polymorphism which is directly related to this disease. First of one is 14 base pair insertion deletion polymorphism. Another one is 3142 GC polymorphism. The 14 base pair insertion deletion polymorphism result from the friendship mutation, that is the insertion deletion mutation. And 3142 GC polymorphism is related to base substitution mutation, where G converted to C. And this polymorphism is very prominent in other autoimmune diseases, including RA also. There are approximately 7 soluble isoforms found. Of them, 4 are membrane bound and 3 are soluble form, which is also associated with this disease pathogenesis. The objective is basically to find whether any association is present between the polymorphism with the soluble HLA-G isoforms in rheumatoid arthritis. To fulfill this object, I collected as well as recruited 84 RA patient and healthy control subject from Malda Medical College and Hospital. The RA patient mainly diagnosed by a specific criteria based on American College of Rheumatology and European League Against Rheumatism. This is the criteria where 4 domain found, including serological data, wound-related data, and other some criteria. We collected 2 to 3 ml venous blood and separated the serum to carrying out the serological test. DNA was isolated by Kaizen extraction kit for genotyping of the both type of polymorphism. In case of 14 base pair insertion deletion polymorphism, PCR SSP method was carried out. SSP signify the sequence specific priming method, and for 3142 GC polymorphism is carried out by PCR RFLP method. As this is a single nucleotide polymorphism, that's we have carried RFFP method because it is a co-dominant marker. And one restriction endonuclease is used, BAEGI restriction endonuclease, which is extracted from Bacillus escheri. And after PCR, the PCR amplified are electropodes in 2% and 3% agarose gel respectively, and gel documentation was carried out. This slide shows the reaction mixture and the primer for both forward and reverse primer, which is related to 14 base pair insertion deletion polymorphism. And this is a sequence specific primer. This slide shows the reaction components as well as forward reverse primer for 3142 GC polymorphisms. Here I show the PCR programming. I standardized the annealing temperature for both kind of genotyping. For first type, the annealing temperature standardized 60 degrees centigrade, and the second type, it is 63 degrees centigrade. After standardization, we perform the experiments. Another one, the soluble HLA-G isoform from in RA patients and healthy control, mainly quantified by a sandwich human ELISA kit, which sensitivity is approximately less than 0.31 nanogram per ml. And there are several statistical software which are used like iPlot, SSP. Now the result and discussion portions. We found that the soluble HLA-G level is significantly low in RA patient compared to the control subset. And the statistical significance is prominent. That means there are some correlation between the soluble HLA-G with the disease pathogenesis in RA cases. This is the electrophotogram diagram which show the exact pattern of band. Like there are three kinds of genotyping are found: 14 base pair insertion, 14 base pair deletion, and insertion deletion genotype. And the band are directly observed and counted for each participants. And this is the another electrophotogram diagram which show the genotyping of 3142 GC polymorphism after specific restriction titration in agarose gel. And this is the table which related to the soluble HLA-G level with polymorphism, but we did not find any correlation between the variables. This is probably due to the small population size, as we included only 84 RAs and healthy control in our studies. But it is very prominently observed that the soluble HLA-G level is significantly low in RA patient compared to the healthy controls. Lastly, the conclusions. This finding directly indicate that soluble HLA-G molecules have its anti-inflammatory properties, and which may be used as a suitable biomarker to diagnose the RA. But this is a preliminary study which does not support any kind of association between the polymorphisms in rheumatoid arthritis. Probably, if we increases the sample size, it would be signified. I acknowledge the ACRB for the financial support and the Medical College and Hospital for providing the blood samples and serum samples as well as demographic and clinical data as well. Thank you. So,
Rajat, this is a very nice presentation. Just I have one small question. You perform statistical analysis. So, do you think the sample size is large enough to have a good amount of idea, good amount of statistics?
yeah, yes, sir. Probably if we recruited large population size, is may significant between the association. Actually, the disease risk factor as well as the odd ratio should be directly correlated. But this study was only for 84 patients and control group. It will not signify any association between the risk factor as well as disease susceptibility.
So, it would be better that if you could increase your sample size.
Yes.
Like that would give you a better perspective of the
Yes,
results that you want. But it's a very nice presentation.
Okay, sir. Thank you, sir.
Okay, Rajat, I have just one suggestion. So while mentioning the statistical software that you have used in material methods, try to include or mention the version, because you mentioned the SPSS. So which version you use, that should be mentioned in material method. So in next time you try to mention. Okay, thank you.
Yes, ma'am.
So, what are
There's some background noise
Rajat, you
coming.
please mute your microphone. Rajat.
Some background noise is
Rajat,
coming.
you please. Yeah.
Yes, ma'am. Thank you. Is it visible, ma'am? Hello? Oh, okay. Good afternoon. My name is Shomir Pavel, PhD scholar, Immunopharmacology and Molecular Cell Biology Lab, Department of Zoology, University of Gurgaon, Malda. My topic is anticholinesterase potential of an ethnomedicinally important edible plant, Padelia foetida. First of all, introduction. Padelia foetida, family Rubiaceae, commonly known as skunk vine, is most commonly used as an edible plant, especially in the northeastern and eastern regions of India. But its ethnomedicinal uses are also undeniable. This plant is reported to have many secondary metabolites that exert biological activities in combating different diseases and conditions like dysentery, pain, diarrhea, constipation, wounds, etc. The present study was aimed to investigate its anticholinesterase, antioxidant, and free radical scavenging properties. Moreover, the plant was also investigated for total flavonoid and phenolic contents as well as lipid peroxidation inhibitory activity. The 70% methanolic extract of P. was also found to possess moderately high anticholinesterase, antioxidant, and free radical scavenging properties. This observation was supported by the presence of a considerable amount of flavonoids, and phenolic compounds. Now the objectives. The objective of the present study is to evaluate the total antioxidant activity of the plant extract, also to assess the anticholinesterase potential, acetylcholinesterase and butyrylcholinesterase inhibitory activities, as well as to assess the scavenging activities of different free radicals that comprises various ROS and RNAs, including hydroxy superoxide, nitric oxide, hydrogen extract of the plant was collected from the local markets, then identified and authenticated. The young leaves washed, cut into small pieces, and shed dried for 7 days. Then the shed dried plants were coarsely powdered and extracted with 70% methanol for about 48 hours. Then the methanol extract was concentrated in rotary evaporator at 40 degrees centigrade to get recipes. And also it was stored at 40 degrees centigrade for future use. Then the using 70% methanolic plant extract, following experiments are performed like total antioxidant activity, hydroxyl superoxide, nitric oxide, hydrogen peroxide, hypochlorous, iron chelation, peroxynitrate, total flavonoid, total phenolic, DPPH, and reducing power activity. Now the results. These are the IC50 values of the plant extracts and also the standards used for the experiments. Here we can see that the extract shows good hydroxyl scavenging activities along with the nitric oxide and hydrochloric acid scavenging activity. In case of hydroxyl, here IC50 value is less than the mannitol, that is the standard use. Also, the extract shows moderately good amount of total antioxidant, total phenolic, and total flavonoid content. These are the graphical representation of different antioxidant activities. This is the hydrogen peroxide, nitric oxide, hypochlorous, and this is of the hydroxy radical. Here you can see that this plant extract is little bit greater than the standard use. That is mentioned. Then this is iron genetic. This is reducing power, superoxide radical scavenging assay. Then here we can see that the plant extract shows acetylcholine and butyrylcholine and lipid peroxidation inhibitory activity at dose dependent manner. From Pearson correlation matrix, it can be said that there is positive correlation between total phenolics and superoxide, nitric oxide and butyrylcholinesterase, and also negative correlation between hydrogen peroxide and superoxide, reducing power and total antioxidant, lipid peroxidation, and total flavonoid. From this principal component analysis, it can be concluded that the butyrylcholinesterase inhibitory activity, total antioxidant, nitric oxide, reducing power, and also acetylcholine inhibitory activity from and hydroxyl radical lipid peroxidation, the total phenolic from the y-axis. That is these are the factors responsible for antioxidant and acetylcholinesterase activity, and which is supported by this PCA plot. These are the research highlights. Padilla contains good amount of total antioxidant, total flavonoid, and phenolic contents. The plant extract shows significant free radical scavenging activities and also possesses moderately high anticholinesterase activity. So it could be good alternatives of synthetic antioxidant in preventing oxidative stress associated diseases as well as could prevent the progression of neurodegenerative diseases. The plant extract work as a better hydroxyl radical scavenger than the standard mannitol at higher dose. The extract also inhibited lipid peroxidation as well as showed anticholinesterase activity in a dose-dependent manner. This principal component analysis of the present study showed that superoxide nitric oxide, butyl polymerisase, inhibitory activity, reducing sugar, reducing power, hyperpluas, and propylphenolics are the most prominent factors that might enhance the antioxidant free radical scavenging and also anti-polymerisase activities of P4. From the present study, we can conclude that, first of all, these in vitro assays indicated that Agaricotida extract is a significant source of natural antioxidants, which might be helpful in preventing the progression of various oxidative stress associated and also neurodegenerative diseases. The extract of P4 could be further explored in the future as a source of useful phytochemicals for the pharmaceutical industry. These are the references. Thank you.
Thank you, Shomir, for your nice presentation. So you have mentioned that you have collected the plants from local market, isn't it?
Yes, sir.
So at which, I mean, what was the season that you selected or collected that plant and then you did all these assays? So it's how many times you have repeated? And my question is, have you studied the, I mean, collected leaves or these plants from the market from different seasonal, different seasons? Have you studied the impact of it?
No, sir. So far, we have collected it from the local market few months ago, but we have also planned to.
That means during summer.
Yes, sir. And we have also planned to study it seasonally.
Because if you study the same parameters and taking plants during winter or in another season, so you can find that there is a lot of variation in the capacity.
So far, we have not done that one, but later we have also do this.
And do you have any? I mean, how you will recommend layman people or common people to consume these leaves? I think leaves are edible, no?
Yes, sir. It is used as green leaves.
So how, because you have studied all the antioxidant activities and other cross scavenging capacities. So what would be your recommendation, or how you can recommend these leaves to consume? I mean, what would be the dose or how many leaves can we?
Actually, these plants are used as green leaves, as people usually take it as boil condition. Actually, cooking, after cooking they consume it. Sometimes it is also used as false medicine, like juice of the plant leaves. So people use it in both forms, in raw form as well as in boiled form.
So my suggestion is you can collect these plants and you can grow this in your garden. I mean, the experimental garden that your university has. So and then you can study these. So that will give, I think, much more reliable data instead of collecting from the local market. So it will give you more reliable data. Okay, thank you, madam.
Yeah. I have a question. So when you talk about the extract, are you taking extracts of the leaves or you're taking extract of the whole plant?
No, ma'am. Actually, it has taken extraction from the leaves, green leaves.
Leaves only. Okay.
After getting dried.
Okay. So, as an extension of this work, do you have any plan to identify what is the active ingredient in the plant extract that is responsible for the its antioxidant activity or the anti-cholesterol esterase activity?
Actually, the main phyto compounds yet not been identified. So, for this purpose,
I didn't get you.
Like
it has already been identified?
No, ma'am, we still not identified the main
No,
compound.
I didn't understand what you said. That's so you could do that, no? That would be a very good piece of work. And then you can compare also whether this active ingredient is more effective, or sometimes even the whole plant extract is much more effective. Okay. Nice presentation. Thank you.
Thank you.
Thank you. Nice.
Hello, hello. My
Yes,
skin is visible
yes, it's audible.
and visible. My skin is
Yeah, you please go through the, I mean, it's visible, but you can go through the slide show is not coming.
good afternoon to all. With a lot of respect and gratitude to chairperson of this session, respected ma'am, sir. Due to some technical problem, I would like to share my mobile screen for presentation. I am Dr. Someshwar Singh, Assistant Professor of Geology, Vivekananda Mahavidyalaya, Bardhaman. In this international seminar, I want to represent a topic that is eco-friendly control of Japanese encephalitis through green silver nanoparticle synthesized from Holoptilia integrifolia leaf extract. Japanese encephalitis is one of the deadly mosquito-borne disease and caused by the virus belonging to the family Flaviviridae. Spreading of Japanese encephalitis has been associated with agricultural development and extensive rice irrigation schedule. According to WHO, more than 3 billion peoples of Southeast Asia and Western Pacific region under the risk of Japanese encephalitis transmission. First major outbreak of Japanese encephalitis was reported from Bankura and Bardhaman district. We, we also, the residents of Bardhaman town, and the main vector of this disease is Culex visnui mosquito. Due to wide range of agriculture, particularly paddy, and the stagnant nature of water, Bardhaman district is one of the prone zone for transmission of Japanese encephalitis, and their vector preferably bead on the fresh water, such as rice field stagnant water. According to WHO, control of vector is one of the indispensable strategy for controlling mosquito-borne disease, as well as control of mosquito at larval stage because they are not spread out and fly to here and there. So target to the larval stage is become more beneficial, is become more controllable for their eradication rather control program. Previously, larvae control chiefly based on application of synthetic insecticides. And we also know that synthetic insecticides such as DDT and lots of things have lot of adverse effect on the environment, on the ecosystems. Those have biomagnification property. Those have ill effect on non-target organism. Non-target organism means those sharing the same habitat of Culex kisnoi mosquito, that is tadpole, that is Diplonicus, that is Chironomus, and other lots of organisms, those sharing same habitat to the Culex visnoi mosquito, as well as pollution of the environment is the main cause, because we can't use today these types of chemical synthetic insecticides. And nowadays, an utmost necessity to develop an eco-friendly or eco-recyclable indigenous plant-derived insecticides that easily biodegradable. And biodegradability nature of these insecticides become more precious nowadays for control of different types of pest as well as different types of vector. Those are the responsible for the transmission of a number of disease in human population. In this research, I found Holoptilia integrifolia. It has lots of distribution in tropical and subtropical countries as well as Indian peninsula. Its common name is chilbil, nata coronja. It's a wide types of medicinal property, anti-cancerous, antioxidants, anti-larval. Number of property of these plants have already reported for Holoptilia integrifolia. And this plant belonging to the family Almaceae. In this research aspect, I use synthesized green silver nanoparticles from this plant as a reducing agent for the control of Japanese encephalitis because it is biodegradable product, it is suitably handleable, and it can be used freely for the control of these types of vector to to stop the propagation of this disease. In this case, we firstly synthesized silver nanoparticles from Holoptilia integrifolia leaf extract. And silver nitrate was used as a salt, and silver nitrate as well as this plant leaf was used for the collection of the nanoparticles. These nanoparticles have characterized by number of procedures, that is UV spectroscopy analysis method, followed by XRD, SAME, TAME, FTIR, etc., etc. And this table indicates the efficacy of this nanoparticle for the control of mosquito pest. Here, 5 type of concentration we mainly used, that is 1.25 ppm, 2.5, 5, 7.5, and 10 ppm concentrations, and show the result for about 24 hours of exposure. In all cases, all 4 instar larvae, first, second, third, and fourth, all were 100% show mortality at this after 24 hours of exposure. When we try to characterize the silver nanoparticles, they show number of property. First of all, we use UV spectral analysis. This is the spectral peak. Here, bioreduction of silver plus ions to silver nanoparticles were done, on which resulting change in the absorption band. And here, UV absorption peak at 421 nanometer clearly demonstrate the presence of silver nanoparticles were synthesized from silver nitrate salt with the help of the plant leaf as a reducing agent. XRD, major peak at 37.92 degree and 44.94 degree agree with the powder diffraction standard data belonging to the silver structures. So it confirmed the purity of the silver nanoparticles. This is the same result, and this is the tame result. Figure 3 and figure 4 show the same and tame figure that clearly demonstrate it is the synthesized nanoparticles, and after measurement, it is 40 to 50 nanometer in diameter of the silver nanoparticles, that is mesocyte. And we also know mesocyte nanoparticles are good one type of controlling agent for different types of vector population also. It is the FTIR results, which indicate the different types of structural components of the nano compounds. And lastly, we show the picture through which different larvae, how they can diet. These nanoparticles particularly target on the midgut, thorax, and gill parts for the killing of this larva. So I think it is one of the best procedure and one of the handy procedure as well as eco-friendly procedure for controlling these types of dangerous disease such as Japanese encephalitis. We know these types of virus can attack at the central nervous system or cerebrospinal fluid and transform a man from 70 kg to just about 25 or 30 kg after the severe effect. So finally, it is one of the best alternative approach for control of the Japanese encephalitis as well as eco-friendly approach for control. It have no any ill effect on ecosystem as well as non-target organism that may be tadpole, Diplonicus, Chironomus, Daphnia, Cyclops, etc., etc. Those sharing the same habitat with Culex bisnoi mosquito. So I think more details research and its marketization must be need for proper application of these types of resource, these types of material for control of different types of mosquito-borne disease. That's all. Thank you to all.
Thank you, Dr. Shingo, for your nice presentation. So I have just one query: why you have selected this particular plant for synthesis of silver nanoparticle? Because there are so many other plants. So have you studied other plants?
Yes, I have already screened near about. I have already done research on this particular plant for about 7 years, and I have already screened near about 200 plant. But these plants show tremendous efficacy of the silver nanoparticle. all plant, majority of plants also synthesize silver nanoparticles by silver nitrile applications with the proper technique. But these plants have, when we isolate the silver nanoparticles by 10,000 RPM and 15 minutes of rotation in centrifuge machines, and the pellet are isolated and weighed according to ppm, when applied it to the field and applied it in the laboratory environment, these plants only show tremendous result except another plant.
Okay,
So
is
out
it
of
cost effective method?
yes, sir, this is cost effective because silver nanoparticles required very minimum quantity.
Yes,
And
yes, it is highly toxic.
if we use that, so it will be mostly beneficial as well as cost effective product also.
Okay. Another question is it effective? Have you studied it's against the malaria mosquito, or have
Yes,
you
sir,
studied
yes, sir. I have published near about. I also synthesize the functional component that we says that we question previous author. I have also mentioned. I also separate proper analysis of that is active principles of the compound.
Okay.
Those are responsible for the control of malaria also.
Okay, okay, thank you. So, any other query?
Yeah. So would I have just one small query that you said that you have screened several plants that you have used for synthesizing nanoparticles, but you found this plant to be most effective. Would it mean that association of silver with some active component with this plant is causing this effectiveness?
Yes,
If
exactly,
it
ma'am.
is so, have you made a comparison between just by using the extract of this plant and an extra nanoparticles of silver extract prepared through using this plant extract as a reducing agent?
Yes, ma'am. I have already done wide kinds of research on this plant. I have already synthesized different extracts from non-polar to polar, from petroleum ether to alcohol. And all these extracts also tested for the synthesis of nanoparticles. But aqueous extract, which in here used for the synthesis of nanoparticles, these nanoparticles have better results than all the aqueous extract. I have also done aqueous extract as well as their synthesized isolation of the active principle for the control of the mosquito larvae.
Now
Another part.
my question is a little different. Yes, but how effective is the aqueous extract of this plant in, in its anti mosquito larva activity? Uh,
I have already
does
shown
this plant extract alone have activity in killing mosquito larva?
plant extracts also have a killing potentiality of mosquito larva, but percentage of killing and at a low ppm is nanoparticles is more far better than plant extracts.
Yeah,
Plant
so
extract require
I
better
think
quantity, more quantity.
such comparison, the data showing these comparisons would also be very useful, very much indicative and useful also.
Already two or three people are at the present situation already communication level. So in this don't I agree this. I have already done these types of research, ma'am.
Okay, thank you.
Thank you, ma'am.
Yes, thank you. Is that visible now? Thank you. Okay. Good afternoon, everyone.
My name is Pradyut Malakar. I am from Kalyani University Department of Ecological Studies, and I am a research scholar of this department. So today my topic is coupling of inline electrochlorination and filtration by co-precipitation method for the supply of safe drinking water free from risk of arsenic. So the arsenic is naturally occurring element which is present in water, soil, food, and everywhere. And this is responsible for making different chronic diseases, human chronic diseases such as organ damage, multi-organ failure, which further leads to death. There are no effective medical treatment for chronic arsenic poisoning. So, in most of the cases, there is Ganga, Brahmaputra, and Meghna plain in Bangladesh and in West Bengal shows maximum arsenic contamination in water, surface water as well as groundwater. Because of that, people of Bangladesh, around 30 million people are suffering from this, and worldwide the number is less than 100 million. And the recent data shows that 70 million people in Bangladesh and 42 million people in West Bengal are affected by this arsenic, out of which 20,000 people per year are dying from arsenic in Bangladesh, and around 1 lakh people worldwide are dying from arsenic in all over world. So according to the limit of Indian limit and Bangladesh limit or Switzerland limit, the lowest value should be less than 50 mg per liter in water, in drinking water. But in case of who, the the lowest value should be less than 10 mg per liter. But except of that, to treat this water, there have several arsenic removal plants in West Bengal, but the scenario is totally poor because according to the recent study, it showed that around out of 570 arsenic removal plant, 475 is not working properly, of which 148 is even in not in still working condition, and the remaining which are in working condition, the out of these 28% arsenic removal plants would not meet the guideline of Indian as well as WHO standard. So therefore, there is a very really need of a feasible arsenic removal, feasible alternative of this arsenic removal plant. So depend on these. So depend on the previous study, we saw that arsenic mainly present in water, groundwater, in arsenite form and arsenate form, which I mean the most dominant in arsenate in groundwater is the form is arsenate that is AS5, which absorb iron hydroxide, mostly the freshly formed iron hydroxide in the presence of strong oxidant. Suppose if we use oxygen, then this oxidation level is little lower, but if we use strong oxidant, then it can be absorbed by those freshly formed iron hydroxide. So even some previous researches also showed that in lower pH and in the presence of iron, the arsenic removal efficiency was much higher than the absence of iron. But when we use, I mean, even in other previous researches, it also showed that when we use iron as well as chlorine as an oxidant, then the arsenic removal efficiency was much better than others. Not only that, even there is any phosphate in the water, then it also always make a trouble for removing arsenic from the water in the presence of iron. So depend on that, we planned to coupling one strong oxidant producing electrochlorination unit with a filtration unit. The oxidant producing electrochlorination unit is a unit was purchased from Watercon, and this unit mainly produce chlorine from the chloride present in water. So there are two electrodes made of titanium coated with deuterium and iridium. These two electrodes are mainly responsible for producing chlorine from the water. and this chlorine is responsible for making this iron, the present iron of water, to form ferric hydroxide, and this ferric hydroxide further adsorb those arsenic 5 form, and which further precipitated in this filter. And the filter media which we used in this case is green sand filter media. So this was the experimental setup where we collect the water from 15-meter well depth, which is around 200 mg per liter was the concentration of this well water. And thereafter, I mean, this is the groundwater, and thereafter we treat this water by the electrochlorination unit, and after that we filter it through the GSP media, green sand filter media, and thereafter we collect this water for further use. And this GSP filter media was also connected with the backwash system. The backwash was responsible for collecting those sludges from these arsenic removal plant. But in the first phase, we had some difficulty because of some iron breakthrough. Because of these, we had to add another green sand filter media filter, so that we can reach up to the highest level of removal efficiency. So this was the system. Here, this is the well, and this is the system which we used for electrochlorination. There also had, in the left side, there also have two filtration unit. So these are the average observation which we received from the system for after the 10 months work, I mean 10-month experiment, which showed that 70 to 80% ammonia removal efficiency of the by the system, and 87 to 99% iron removal efficiency, 76 to 94% arsenic removal efficiency, where 200 mg per liter, I mean on an average, 186 mg per liter was the arsenic concentration, which reduced up to 28 mg per liter. That is lower than the Indian standard, but little higher than the WHO standard. Even in case of manganese, also we showed we succeed to receive 55 to 96% removal efficiency. And we also previously we discussed about phosphate, because phosphate is a inhibitor of arsenic removal or arsenic co-precipitation. But in that case, we also achieved to remove 75 to 81% phosphate from the water, from the contaminated groundwater. By this technology, by this combination of those technology, we succeed. This was the inlet water arsenic concentration, which was from 150 to 350. This was the inlet water arsenic concentration, which reduced up to which was always in maximum cases. This was lower than the Indian standard. Sometimes we also receive the value lower than WHO standard. In case of iron concentration, we also, because iron here we used as a co-precipitator of arsenic. So with arsenic we also remove the iron, and in that case also we also achieve the Indian standard limit. In case of manganese, the manganese removal efficiency was also very high. That was almost 96% in maximum cases. Sometimes also receive a little bit lower in case of second experimental phase. They had some difficulty to remove manganese, but except that we always achieve almost 96% capacity of removing arsenic manganese from the water. Not only that, we also achieve to remove turbidity from the water by using this technology. Here, the main system was responsible. That is, I mean, in the lower pH, I mean, from 7 to 8, within this pH, the system produced maximum oxidant, which increase the Warp, and this Warp is responsible for making this iron to iron hydroxide and to form arsenic 3 to arsenic 5. So whenever we achieve this, I mean, we were able to get this breakthrough chlorination breakthrough. We also achieved this 90% arsenic removal efficiency. Even in case of manganese and iron also, when the Warp was higher than 300, we also achieved the maximum efficiency of removal of manganese and iron. In case of ammonia, the inlet water had almost more than 1 mg per liter ammonia concentration, which reduced up to 0 to 0.5 after treatment. Orthophosphate, I already had discussed about that 75 to 81% orthophosphate was removed from by this technology. So the main finding of this work is the present study findings could provide a considerable significance of the use of solar-driven electroplurination unit was mostly capable of reducing arsenic from the groundwater. So this technology is not only cost effective, but it takes very less effort of operation and maintenance. I mean, this technology is fully monitored by online configuration, so we don't have to go each and every day for maintaining this technology. This kind of technology is mostly significant in those rural areas where electricity is not available, but land is available in large quantities. The co-precipitation process was always effective to reduce arsenic concentration lower than the permission level limit when iron is present in the water as a co-precipitator. Removal of phosphate is always important because without removing phosphate, it's very difficult to remove arsenic from the water. Thank you.
Thank you, Pradut. It was a nice piece of work. If somebody else has any question, we can go ahead.
I have no question.
So Pradut, you have mentioned this is not a cost-effective method. So you are working on this so that it to make it more cheaper, or I think you are working on this, or what is your idea how to make it more convenient and it will reach the village people?
No, actually, sir, it's a cost-effective method.
Okay,
I mean, it takes very less amount of money.
I mean, what's the running cost? I mean, if you can install it in a very remote area, so is there any reusable part or any column that you need to change after certain time like that?
No, actually, the system is fully automatic. So maybe once in a month, or more than that, maybe 6 months. After 6 months, we have to change the filter media or sometimes was the media, because the media which we use that is fully backwashable. But sometimes there is a clogging if there is a huge amount of iron is present in the water.
Okay, so in that case, some pre-iron remover can be installed.
No, sir. Actually, if we use those pre-iron removal, then the iron will reduce the capacity of absorbing those arsenic.
Okay. Thank you, Pradut.
Thank you, sir.
Hello.
Yes, sir.
Hello.
Yes, sir.
So finished? No. Now discussion session is going on. So no presenter there.
I think we have come to the end of this session.
So I am Dr. Bhakta speaking.
Yes.
Professor Jayed sir.
Yes.
Thank you very much for your gracious presence in our conference.
Thank you for
and
inviting
to
us.
all of our participants. Now we will move to the track 1. I request to all participants, person and speaker. Yes. So I also Professor Rita Ghosh and also Professor Angsuman sir, I request you, and also I thank you from my core heart for your gracious presence in the conference and giving your effort as a chairperson. So again, thank you, sir and madams. I also invite you, all of you, to present in the track 1, where we will conduct the valedictory session very soon. So please be present there for our this program.
Thank you.
Okay, thank you everybody. Now,
but would it be
yes,
another link? Thank you, Dr.
Yeah,
Bhakta, for your invitation. And it was my
Yeah.
pleasure to be present during this session. So you are
Okay, we will
asking
send.
for
We
us
will
to be
send.
present in. Oh.
Ma'am,
It's another.
the link I am sharing with you, sir, ma'am.
Okay.
Okay, thank you. We'll share
Thank
now.
you very much.
Thank-
Yeah, thank you.
Yeah, we'd like to thank all the speakers. It was a pleasure to be present here. Everybody presented very well. Thank you.
Thank
Okay,
you very
thank
much.
you very much, ma'am.
Thank you. Thank you. Thank you.
Okay.